Hello. Ajay. Hello. Ajay. Voice clear ಇದೆಯಲ್ಲ? ವಾಯ್ಸ್ ಕ್ಲಿಯರ್ ಇದೆಯಲ್ಲ? ಕ್ಲಿಯರ್ ಇದೆಯಲ್ಲ? ವಾಯ್ಸ್ ಕ್ಲಿಯರ್ ಇದೆಯಾ?
ಕ್ಲಿಯರ್ ಆಗಿದೆ ಸರ್.
ಓಕೆ ಓಕೆ.
ಅಲ್ಲ ಇದು ಸಾಕಷ್ಟೇ, ಅದಕ್ಕೆ ಸ್ವಲ್ಪ ಹೊರಗೆ ಹೋಗ್ಲಿಕ್ಕೆ ಮಾಡಿದ್ರೆ ಆಟೋ ಹೋಗ್ತಿದ್ದೆ. ಹಲೋ 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 ಕೇಳ್ತಲ್ಲ ಹೊರಗಿದ್ದಾರೆ ಹಲೋ
ಸರ್
ಮೇಡಮ್ ಕೇಳಿಸ್ತುಂಟ ಮೇಡಮ್
ಕೇಳಿಸ್ತಿದೆ ಸರ್.
ಒಂದು ಸ್ವಲ್ಪ ಮಾತಾಡಿ ಮೇಡಮ್.
ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ.
ಓಕೆ ಕಂಟಿನ್ಯೂ ಮೇಡಮ್.
ಸರ್ ಎಷ್ಟೊತ್ತಿಗೆ ಶುರು ಆಗ್ಬೋದು ಕಾನ್ಫರೆನ್ಸ್?
ಒಂದು 30 ಮಿನಿಟ್ಸ್ ಅಲ್ಲಿ ಸ್ಟಾರ್ಟ್ ಆಗ್ತದೆ.
ಇನ್ನು 30 ಮಿನಿಟ್ಸ್.
ಓಕೆ
ಸರಿ ಸರಿ ಆಫ್ ಮಾಡಿದ್ರು.
ರಿಫ್ರೆಶ್ಮೆಂಟ್ ಇದೆ ಅವಾಗ ಬೇಕಾದ್ರೆ
ಸರ್ ಆಡಿಯೋ ಪ್ಲೇ ಆಗ್ತಾ ಇಲ್ಲ.
the scientific deliberation, scientific activity. Friends, a day was there. Once upon a time, when there was no technology, during the time of Lamarck, during the time of Mendel, Darwin, Weizmann, etc., etc., how they used to publish, how they used to popularize their research and investigation. ನಿಮಗೆ ಗೊತ್ತಿದೆ, ಊರಲ್ಲಿ ಹಳ್ಳಿ ಮರಕ್ಕಾಗ್ರೆ 4 ಜನ ಕೂತ್ಕೊಂಡು ಪಂಚಾಯತಿ ಮಾಡ್ತಾರಲ್ಲ, ಏನೋ ಡಿಸ್ಕಸ್ ಮಾಡ್ತಾರೆ, ಅದ್ರಲ್ಲಿ ಏನೋ ಸಮ್ ಔಟ್ಕಮ್ ವಿಲ್ ಕಮ್. ಆದ್ರೆ ಅದು ಸೈಂಟಿಫಿಕ್ ಆಗಿ ಆಗೋದಿಲ್ಲ, ಅದು ಡಿಸಿಪ್ಲಿನ್ ಆಗಿರೋದಿಲ್ಲ, ಅದನ್ನ ಡಾಕ್ಯುಮೆಂಟರಿ ಎವಿಡೆನ್ಸ್ ಆಗಿ ಕನ್ಸಿಡರ್ ಮಾಡ್ಲಿಕ್ಕೆ ಆಗೋದಿಲ್ಲ. ಅಂಡ್ ಇಟ್ ವಿಲ್ ನಾಟ್ ಬಿ ಎನ್ ಎವಿಡೆನ್ಸ್ ಅಟ್ ಆಲ್. ಅದಕ್ಕಾಗಿ a group of scientists, researchers, investigators, thinkers, academicians grouped together, they interact themselves, discuss the pros and cons, and then they will make a resolution, and later some findings will be recorded. That is the evidence. And that type of evidence, I request Professor Muthuchalian sir and Dayanand sir to kindly occupy the chair. So therefore, in the scientific sessions, to make it an evidentiary document, there should be an assembly, there should be a gathering, there should be a presentation, there should be a question and answering, there should be some kind of doubt clearing. Then that particular research, that particular finding, investigation is approved or granted by the group of academicians, researchers and scientists. Therefore, the technical session is the most important session for every seminars. Most of you have come with your poster presentations, oral presentations. Most of the budding students are there. Nowadays, in the national education policy, in BSc itself, the project work is started. So it is implemented. Therefore, kindly attend, every one of you attend seriously. You have been given the conference kit with the notepad and pen. Wherever you have doubts, wherever you have some clarifications required, please note down. You can ask. And then you can improve your quality of research. Some of them will do research, but they don't know how to publish it. So you will come to know all these things. So I request each and our, the money we are spending, the time we are spending, the energy, the risk we have taken, everything is for the benefit of our youngsters. There is nothing is going to be useless here. Everything is useful. Great scientists are here. Tomorrow also some of them are coming. Afternoon some of them are joining with us. So I request each and every one of you to kindly be here and make use of this opportunity. Friends, before we go for the keynote address by Honorable Vice Chancellor Professor Dayanand sir, we have a video sent by our Honorable Vice Chancellor Professor P.S. Edapudi sir, and I request the technical committee to broadcast this message. After this, I request Professor Muthuchalian sir to be the chairman of this keynote address. because to have any interaction with anything. There is no question answering session for the keynote address. Keynote address is an bird's view in the subject, in the field, and opportunities and potentialities, future, what is done, what is to be done, what would be the benefit, everything. It's a bird's view. So there will be no question answer session, but I request Professor Muthuchelian sir to be the chairman of that keynote address. Thank you.
Thank you, sir. Optimism is the faith that leads to achievement. A very good afternoon to everyone gathered here. I would like to extend a very warm welcome to all of you for the much anticipated technical session of the National Conference IMTSF 2022, Integrated Microbial Technology for Sustainable Future. This session will surely give us a lot of learning about different microbial technologies for a better world. I request the technical team to play the presidential talk of Professor P. Subramania Yadipaditaya Sir, Honorable Vice Chancellor, Bangalore University.
Very good. Greetings from I'm extremely happy to know that our university, the Department of Studies in Microbiology, PG Center, Janakaveri Campus, Madikeri, Kodagu, in association with Microbiology Society India, PEARL, a foundation for educational excellence, are jointly organizing 2 days national conference on integrated microbial technology for sustainable future. I am very happy to note that the chief guest and my friend, Professor Dayan Dagsar, Honorable Vice Chancellor of Gulbarga University, will be delivering the keynote address. The guest of honor, Professor K. Muthuchelian, former Vice Chancellor, Periyar University, Salem, will also grace the occasion. And this is a very timely and topical theme for this 2-day national seminar. And there are also technical sessions and distinguished resource persons like Dr. Parli Venkateswaran Bhaskar from Goa, Dr. Anirban Chakrabarti from Mangalore, Dr. Prakashyam Halami, Mysore, Professor A.P. Lipton from Kanyakumari, and Professor M. Haridas from Kerala. And Dr. Ashok V. Bhadkar from Pune. And there will also be oral or poster presentations by Dr. A. Jayakumar Nair from Kerala. In that sense, it is truly 2-day national conference, both in letter and spirit. I congratulate and commend the efforts of Professor M. Jayashankar and his team, especially Dr. K. S. Chandrasekharaya, Director in Charge, Dr. B. S. Gunashri, Chairperson of the Department, Dr. I.K. Manjula, who is also in the reception committee. I request all of you to make the best moments of truth and actively participate in the deliberations and discussions of this 2-day conference. Thank you very much for your time and opportunity. Best wishes from Mangalore University. Jai Hind, Jai Karnataka.
I welcome Professor K. Muthuchelian Sir to chair the keynote address session and Professor M. Jayashankar Sir to be the rapporteur. I welcome Professor Dayanand Akhzar sir to deliver a keynote address on the topic Microbial Diversity, Technology and Bioactive Molecules for Human Welfare.
Good afternoon to all of you. Esteemed chair and my best friend, Professor Muthu Chalian, my good friend and the organizing secretary of this event, Sir M. Jaishankar, all esteemed faculty members, very distinguished speakers on the occasion, dear participants. Before I start presenting on my topic, I would like to say two issues to the chair particularly. So though it has been informed that there won't be any questions, but still as a speaker, I love to have the questions from the audience. But please permit me to have the questions during lunch hours. So that I'll be getting the benefit of the time also. Secondly, though I have been permitted by the organizer to speak for about 30 to 45 minutes, but let me have a clearance from the audience how patient you are to listen to me for how many minutes. If you want to speak to me, I'll speak only for 10 minutes, or 30 minutes, or 45 minutes, not beyond that. Can I talk to you for about 30 minutes or 40 minutes? Fine, sir. And somebody has to remind me and you have to stop me. I know that we are basically teachers. It is difficult for us to stop, particularly when we start talking on our own particular topic. That too, most fascinated research in which we have put in the service of about almost 30 years. With the topic as such, integrated microbial technology for sustainable future, I tried to compile my research which has been started from 2001 onwards till date. 2001, that is the period in which we have started working with a special group of microorganisms, most neglected group of microorganisms, so-called actinomycetes or actinobacteria. I thought how actinomycetes research, which was carried out right from 2001 till date, can be compiled with justifying the conference title. Now, the broad title is something like Microbial Diversity, focusing on the diversity of actinobacteria, and then bioactive molecules, covering an enzyme, a pigment, and a biosurfactant, just to make the different fields of applications from the technological point of view and how these are being used or served in terms of technologies to serve the mankind. So that is the whole theme of my presentation for about 30 or 40 minutes. Next slide, please. Biodiversity. I think nowadays students can have access to the wonderful information that is available in the internet, right? But still, it is always essential for each one of us to understand some basic definitions before we start understanding the topic of our own presentation. So biodiversity, that is the variety and variability of living organisms and the ecosystem in which they occur. If you look back into the history of it, I'm not going to explain to you. With the biodiversity aspects of way back during 1992, the concept has been started. And you know what the importance of biodiversity is today in general, focusing on microbial biodiversity, which I would like to put it as only microbial diversity. And if you look into the exploration of biodiversity as sustainable development for the welfare of the mankind, which is at most important. Just look at these two world scenario where the diversity is playing an important role. And my focus is only from this point of view. The whole India has been considered as a hotspot for the biodiversity from variety of the living beings, from which I'll be focusing only on the microbial diversity. Next slide, please. As I was referring, the microbes or microbial world is which is most important because we cannot visualize with our naked eyes. We can visualize only the group of organisms in the form of the cultures or the colonies, but we never realize the organisms because of their size, right? And if you look into this, Martin Alexander's statement that makes us to realize how critical the role of microorganisms in the field of science and technology, focusing the welfare of the mankind. I would like to just state here, if microorganisms were not there, the entire life on the earth would have been chopped to death a long back. Origin of life, evolution of microorganisms, major milestones achieved, and major significant contributions made by varieties of the scientists in the development of the microbiology can be realized under this statement. Microorganisms constitute a huge and almost unexplained reservoir of resources likely to provide innovative applications. It is an ever-encouraging field from the nature to look into the microorganisms. As far as the microorganisms are considered, it has been estimated that about less than 6% of all microorganisms on Earth have been studied. My dear students, you'll be surprised if I say that it is an universal estimation that though such a vast amount of the microscopic research has been done across the globe, it is not even tip of the iceberg has been explored in terms of microorganisms. It is not even 6% of the total microbial population has been explored by the mankind. Imagine what an amount of the strength or the potential of the microorganisms is still there in the nature, in the earth, in the universe. Next slide. Coming to the microbial diversity, as I said, India is a hotspot which has been considered across the globe. But my focused area of research from a particular point of ecosystem or the ecology. That is, I would like to draw your attention towards the limestone. What is the limestone? A major raw material which is normally being used for the production of the cements. Major raw material. If you look at the history of the production of the cements, naturally all the cement factories of the industry are being located in the area in which limestone is a basic raw material which is available in that particular geographical area. Fortunately or unfortunately, my jurisdiction of the university lies in such a hotspot where we have the entire Deccan Tract or the belt of the limestone quarries or the limestone in the geographical situation where we have used large number of industries for the production of the cements. We have more than a dozen of industries contributing the total required 70% of the cement throughout the Asian countries. You can imagine what an amount of geographical potential is there in that particular geographical area. The question is, being a microbiologist, why I am attracted towards these particular limestone quarries? Go to the next slide, please. If you look into that, naturally as a basic microbiologist or a microbial ecologist, so some of the answers we need to find for these kind of statements or the questions. Habitat, ecosystem, microenvironment, niche. Straight away I'll be coming to the niche as such. What is a niche? It is not just an habitat. Ecosystem is a much broader area. Microenvironment is the conditions which are prevailing in that particular area. Please remember, habitat, ecosystem, or the microenvironment, all these constitutes in a given particular geographical area, a small selected area where microorganisms try to prevail themselves, exist themselves, survive themselves, perform themselves in those given conditions. That's what we try to put it as a niche of microorganisms. So ultimately, having known the basic understandings of these ecological principles of the microorganisms, we thought, can we explore these limestone quarries or that particular kind of a microenvironment or that particular kind of a niche wherein we can look for the prevailing predominant population of the microorganisms. That's how our area of interest in the research has started way back during 2001. From that onwards, next slide please. Again, microorganisms are very wide group of the living beings, prokaryotic and eukaryotic microorganisms. Basically, you can put it as 5 or 6 or 7 groups of the microorganisms: bacteria, fungi, actinomycetes, algae, viruses, protozoans, so on and so forth. But the question is, as I was telling in the morning, the most neglected group of microorganisms are a unique type of prokaryotic microorganisms which comes in between bacteria and the fungi. That is the actinomycetes. Now we also put them as actinobacteria. So why we have attracted towards actinobacteria? Please remember, it is not that we are interested. It is not that we are interested to focus on research with actinobacteria. It was just coincidence. I'll be explaining that in some of the slides later. But I want to make you very important that why actinomycetes are so important, because of quickly, if you look into this unique group of prokaryotic microorganisms, which carries both bacterial and fungal potentials, and then greater metabolic diversity, higher adaptability to extreme conditions. Imagine temperature as much as about 50, 60, 70 degrees centigrade. Actinomycetes are bound to there. Imagine the pH, right from the acidic to the extreme alkaline, even up to 12 pH, actinobacteria are there. You can just imagine their metabolic diversity. They are capable of carrying out or metabolizing any kind of the nutrients they come across, despite of the typical microenvironmental conditions, particularly focusing from the point of the temperature and pH. I hope you will realize that temperature and pH are the two most important physio-ecological conditions in which every organism tries to survive in nature. That has made the actinobacteria most potential groups when we compare with bacteria as well as fungi. And that is quite possible because of the high GC content, about 8 MB. Even if you take the genome of the E. coli, which is, I leave it to your imagination, then you can imagine it is almost 3 to 4 times larger than the E. coli, which is a universal example for the study of the microorganisms. That is the kind of the complex potential genome which is present in majority of the actinobacteria, which makes them very potential, promising source of wide range of important enzymes, relatively less explored for other than antibiotics. Yes, we do accept the moment we say the microorganisms, which reminds us of Fleming Alexander with the antibiotic. What is the antibiotic he has given? Alexander Fleming? Penicillin. Is it penicillin? Okay, whatever it is, I leave it to you again. The moment we say actinobacteria, actinomycetes, they are well known from the point of antibiotics. And our approach was entirely different. We never wanted to touch the antibiotics, but something else, either in the form of the other secondary metabolites, including the enzymes, pigment, and also a biosurfactant molecule. Next slide, please. Next slide. Yes, next slide. Straightway, I'm emphasizing only the few important slides. I have as many as 50 slides probably, if I am right. Even if I speak 1 minute, it takes 50 minutes. So I want to skip some of the slides, which are very, very basic for all the students to realize and understand the topic of my presentations. So you can just imagine. So these are the various group of researchers. Please remember, I'll submit the whole credit of my research only to my research scholars. Absolutely, I have not done anything. Absolutely, I have not done anything. The whole kind of research is being carried out only by the students, except scolding them, very rarely guiding them, putting them on the track in a right direction. That's the kind of a job what I have done. We could achieve all these things, including great publications and also patent. So that is the message I wanted to give it to the students. Please remember, it is the you young microbiologists who can do wonderful research, provided if we have a bit collaborations with the guides from only the point of preparedness to get scoldings from the guides. Preparedness, getting scoldings from the guides, from the academic point of view. And then be prepared to work very hard under the directions of the research guide. And you know very well that, yes, research guides cannot do any kind of physical work or the table work along with the students, but still they command that in anticipation of the significant contributions only by the research students, not by anybody else. You just remember, all these students, they're all well placed. So right from 2001, we started working on the actinobacteria. We used to go to the fields for the collection of the samples, and these are the different habitats you can see, what we call them as limestone quarries. If you go to the various fields, what we call them as the quarries, anything that is being explored from the geographical areas, they left behind in the concerned areas hundreds of years together. And a typical microenvironment develops in that particular queries. And then we were interested in getting the samples from such microenvironmental conditions and then assay them, analyze them for the actinobacteria. You can see he is the scientist right now who is heading the division of NCIM, NCL Pune, National Council of the Industrial Microbiology, being headed by Dr. Syed Dasgir, who is heading. Remember, he is the first person from his laboratory to detect coronavirus in the entire country, and it's a leading laboratory. It's a leading laboratory of the CSR institutions in the country. Please remember, I'm very proud to say that he was the student in our lab who has initiated the working on actinobacteria. That's how he has visited to the field and started collecting the samples. And these are the only two differences I would like to make. They are the limestone quarries, and some other part within the same geographical area, we have a lot of natural soils which are whitish in color. They're called as lime powder natural deposits. Those who are interested, if you visit sometimes any Deccan Trough areas, including the part of the Andhra Pradesh and Karnataka belt, you can make out these kind of limestone quarries and also natural heap of lime powder. So they were our target areas for the collection of the samples. Next slide, please. This continues. We have a very colorful, beautiful geographical areas when we start extending our Deccan Trough from Karnataka to the Andhra Pradesh, including Kurnool, Betamcharna, so on and so forth, up to the Anantapur. There are the various geographical conditions where we started collecting the samples. Next slide, please. Now it is stated by very simple. Any textbook would give this information. How do you isolate the organisms, particularly the actinobacteria or the actinomycetes? I just written some of the important features, some of the important conditions in which we can isolate the organisms. I don't want to explain all those things. Please remember, what is important is some of the culture media, most important, some of the culture conditions in which we can expect the proficient growth of the organisms, in which we must be in a position to distinguish from the bacteria and fungi. I told you that actinobacteria are the most potential intermediate organisms between bacteria and fungi, and we need to distinguish them. Next slide, please. You can see the colonies. A simple starch cation agar, applying those typical pH, temperature, and incubation period, we could get the colonies. Remember, a typical colony characters which can be distinguished from the bacteria and also the fungi. Fungi which are filamentous long colonies. Of course, they are pigmented or highly colored one. Bacteria are normally small to medium sized in nature, which are mucoid in nature. Unlike these, actinobacteria are highly dyed colonies, wrinkle colonies, hard colonies, powdery colonies, and highly pigmented colonies. Those who are interested, you can get back into the literature and try to understand. And they are the actinobacteria. And another term which has been coined way back during 1990s as rare actinobacteria. Among the actinobacteria, there are few actinomycetes or actinobacteria which are very, very micro in size as far as the colonies are concerned, which have been placed as rare actinobacteria, which are playing a typical role from the point of their secondary metabolites, which we'll be speaking in some of the next slides. Next slide, please. They are the pure cultures, simple pure cultures. Now, look at this. A change in the research path. At the beginning, I told you we never interested in the actinobacteria. Like any other team of the researchers, like any other teachers also, myself and my students were more interested in getting the PhDs within the shortest span of period, submit thesis, get the award, of course, with 1 or 2 publications. Naturally, we were concentrating on the bacteria and the fungi. The topic with which we started is we wanted to explore color-producing microorganisms. So our basic objective was isolate the organisms which are producing the best edible colors. We started with that. And incidentally, when we could isolate on a starch casein agar from the samples of limestone quarries, the beautiful colored colonies we could see. And unfortunately, I'm using the term unfortunately because we were never interested in the actinobacteria. We know that cultivation of actinobacteria takes minimum weeks together for the incubation. Observation takes months together. Confirmation takes years together, which is not an easy task. Neither teachers are prepared nor students are prepared because we are interested in only the instant results, getting our PhDs and publications in the earliest short span of time. But unfortunately, what has happened is all such beautiful pigmented colonies, what have been selected as a pure cultures, they turn out to be the actinobacteria. They turn out to be the actinomycetes. They turn out to be the Streptomyces genus, belonging to the varieties of the species. And we are compelled to start working on that. So my whole research is by compulsion. Is by compulsion, not by the interest. If you look at the standard definitions given by the varieties of the leading reputed international scientists, research is always accidental, incidental. If everything is being done as per the standard protocols, you get the expected results, you get the beautiful publications, you get the patents, but please remember, you can never get the breakthrough research. You can never get the breakthrough research. If it is accidental, if it is incidental, certainly we are bound to get some breakthrough research. That's how our journey starts from the next slide onwards. Next slide, please. With those pure cultures, next slide. Yeah. Simple compound microscopy features. Students started working on those pure cultures. Compound microscope with 100x objective, oil immersion. I don't have to explain to all of you. Please remember, those who are not perfect in these basic microscopy techniques, you shall never be a microbiologist. You can do wonderful things with all advanced software technologies. But if you fail to do a simple staining of the microbial cultures, you can never be a good microbiologist. Keep that in mind. You must be well versed with operating the simple compound microscope, light microscope with 100x objective, or putting the oil immersion also. So that's how, next slide please. That's how, look at their filamentous structures, how do they appear. And then next slide please. The same cultures, when you subjected for the electron microscopic features of their sporulation characters, they are the typical characters. Please remember, the great literature has been well established for the identification of the actinobacteria right from the based on the pigmentation and also the sporulation features, straightway based on the colony characters and the microscopic characters. Very easily one can assign that organism to a genus level. That's how I said straightway all the organisms what have been isolated by the students from varieties of the samples collected from the limestone quarries, all have been belong or assigned to a genus Streptomyces. So our journey starts from the Streptomyces. Next slide, please. We are compelled to do that. Biosystematics. If once we have the cultures in our hand, we know that to what genus it belongs to, we need to carry out further. And please remember, this is the stage where all those pure cultures which were belonging to the varieties of the colors, we had a publication in ARBEC, Asian Review of Biodiversity Journal. Out of curiosity, just based on the varieties and the colors of the actinobacteria that have been isolated, we could publish it. On the cover sheet of that journal, all these 6 pure cultures have been printed, which was attracted towards so-called leading actinobacteriologists working elsewhere in the different parts of the world. And fortunately, This period or that paper was in the hand of Wen Jun Lee, who is working in China, who has been called as or referred as father of present actinobacteria. Luckily, he could look into those colonies and then straight away he could contact our laboratory, and he has invited the student who has just isolated the actinobacteria from those limestone quarries. Please remember, it was with full scholarship. Student from Gulbarga University of Microbiology Department has been invited by the Chinese government by Wen Jun Lee to carry out further advanced research beyond our imagination, What I'm trying to tell you is, if you are interested linking yourself through some breakthroughs, certainly any microbiologist can take up your research to a greater heights. Right? So then we started looking into its entire systematic classification, biosystematics. We put it as a basic biosystematics, then we go for the molecular systematics, then we go for the varieties of the chemical components that are present in the cellular structures on the cell membranes or the cell wall, what is being called as chemosystematics. With these three, we try to nomenclate them, name them, publish them. That has taken us for almost 7 to 8 years. Quickly, let me go through the next few slides, for which students have taken almost about 7 to 8 years. Next slide, please. Molecular systematics, isolation of genomic DNA. Let me not put into all these things. You are much better than any of the present researchers. You can have access to your online and all the recent advanced data. You can just have the standard protocol and subject your cultures through all these techniques. Please only remember what are the important analysis components one has to do it before you go for the publication of your so-called a novel isolate or a new isolate. Next slide, please. Yeah. Chemosystematics. Please remember, go to the back side, previous slide. Yeah, that was the end of the molecular systematics, where all these 6 isolates have been analyzed for the 16S rRDNA and then gene sequencing, then referring to the existing genome bank, try to give as a reference position in the phylogenetic plate. And this is the stage where we wanted to publish the paper. We have sent this paper. Please remember, with all that work in the Chinese Institute of a leading actinobacterial research laboratory, it was rejected straightaway way back in 2007. Do you know the reason? That was almost the period wherein any novel isolates, if at all they have to be published and reported, they shall be subjected for the analysis of the chemosystematics, the important chemical components with which they differ themselves with the existing strains, not necessarily only the basic, not necessarily only the molecular systematic. Chemical components which are present either in the cell profile, cell wall, cell membrane, has to be reported, and then their distinct features with the existing isolates has to be reported, then only they would be accepted for the publications. So we are compelled to go for the chemosystematics, where we try. Next slide, please. We have analyzed for the cell wall amino acids. Next. All the isolates were analyzed for the cell wall fatty acids. Next. Polar lipids. Please remember, they make a significant difference as far as the novel isolates, with special reference to the Streptomyces. We know Streptomyces is the most notorious genus amongst the microorganisms, particularly amongst the actinobacteria. There are thousands of species within the genus Streptomyces, but please remember, it is only these cell wall polar lipids which are present in the cell wall of that particular Streptomyces make them very distinct with the existing isolates. Thereby, we can assign it as a novel or a new species. So that's how polar lipids played an important role. Next, please. Whole cell protein profile, yes. Next. Yeah, with that chemosystematics, you may not imagine in a place like Gulbarga, and never mind to state that it was one of the most rural area in which university has been located in Karnataka, which is in the north part of Karnataka. Now it is being called as Kalyana Karnataka, which is totally rural area. Majority of the students comes only from the rural area. Please remember, with a pure heart and pure mind, where they can work for the research. Receiving all this schooling from the guide, still they are interested. That's how a student, Dr. Syed Dastgir, hailing from the rural area, coming from the small village, who is leading the country in an CSR NSL lab. It is he who could go to the Venjun laboratories, could carry out these molecular systematics, chemosystematics, and we could report 1 new genus and 6 new species of Streptomyces. I just quickly go through those organisms, their name and the publications. The very first one was Actinorhextispora indica, a new genus, first time from the Indian soils. I don't go into the details. You have all these papers available online. You can refer at any point of time. Next, please. Streptomyces gulbargensis. Naturally, Gulbarga is a place where we worked on it. And when we wanted to give the name of Gulbarga, and the earlier trend was anybody who used to report a new species or a new genus, normally we used to give the name of the guide or the student. Naturally, I was also interested. Why it cannot be in the name of Dayanand? Why it need not be a name of student in combination with the guide? Remember, that is the time, unfortunately, universal standards were established for giving a nomenclature or a new name to any novel species. They shall be named only after the institutions, the area in which it has been isolated, or any unique potential functions because of which they have been proved are going to be proved as a novel isolate. Yes, we felt. Let me name it place from where my university is hailing. The very first new species that was published from the Gulbarga soil was named as Streptomyces gulbargensis. You can see that is the paper which has been accepted in the, published in the Anthony von Leeuwenhoek. Now there are only 3 journals across the world which are meant exclusively to report only the novel species of the new strains. And all of them, fortunately, unfortunately, lies in the Germany. So they are the leading journals which publish only the new species and the new journals. Right, next species. Streptomyces tight tolerance. I said tight tolerance is, this is the organism which is tolerant to 3 important physiological features. Highest pH, highest temperature, highest salt concentration. We have no other isolates in the globe which is going to be tolerated for all these 3 important physiological features. Hence it has been accepted as a new species. Next one, please. Allostreptomyces indica species, novel, isolated again from Indian soils. Next one. Streptomyces deccanensis, because my aim was to name to the habitat from which they were isolated. Deccan Trap was a long region starting right from the Gujarat, leading up to the or ending up to the Andhra Pradesh coastal regions, where it is being called as a trap, which is a typical limestone raw material which is existing in the earth crust, that is in the geographical conditions. So we named that organism as Streptomyces deccanensis. Next, please. Nanomedia Indica, again novel species, novel actinomycete isolated from the limestone, open pit mine from India. Next, please. These are the only organisms other than Streptomyces which was belonging to the East. So out of those 6 pure cultures, what has been shown earlier, this is the only, out of 6, this is the only organism which was belonging to the Saccharomyces. But anyway, we could not touch it further for its exploration. Right? That was our publications, led by more than half a dozen of students who are worked only with the actinobacteria. Now, majority of the students right from 2001 were working with only actinobacteria in our laboratory, which is named as ADBT laboratory. The grants with which it has been established, the topic or the focus area of research is actinobacteria, diversity and bioprocess technology. That's how lab has been nomenclated. And quickly, from this diversity point of view, I want to take you to the bioactive molecules. Next slide, please. I have written it here as a mandate by DBT. Then we had a project from the DBT, DST and UGC. Then with a great hope, we went for submission of a proposal to the DBT asking for the further grants. Then they said, why are you bothering about the novel isolates or the new species? Whether you put it in your laboratory or in the nature, they are there in the Indian soils. Why should you be bothered on those things? India is looking. National objective is entirely different. If you're really a true microbiologist, try to explore the potentiality of such organisms which are there in the nature, what you're telling them as a novel isolates. Then that was a mandate from the advisory board by the DBT. They said, no more isolation of the novel species. Whatever the isolates you have in our laboratory, right in our hand, try to explore them for potential applications. And then we could change our directions of the research. We start exploring or exploiting these novel species for the varieties of the bioactive molecules, where we started with not the antibiotics. We wanted to see, is there any potential with these organisms from the point of the a pigment, an enzyme, or a biosurfactant molecule. We have varieties of almost 2 dozens of students who started working only on those lines, exploring these, and we started with tyrosinase. Incidentally, we could have a literature. We are looking for an enzyme. Tyrosinase is an important constituent enzyme, which is going to be produced out of the metabolizing of the tyrosin, and it has got a very typical. If you cut apple and keep it for a while, if you cut brinjal and keep it for a while, if you open up banana and keep it for a while, exposed to the air, it turns itself into the brown color. If you understand the scientific reason why it turns to the brown color is the role of tyrosinase enzyme, a kind of the tyrosin which is present. When it gets exposed to the atmospheric air or the oxygen, then a redox potential enzyme plays its role, turns out to be the brown color. That's what we wanted to explore it from the point of the metabolic activity or the enzyme activity of these actinomycetes. We have screened them for the tyrosinase. From then onwards, our path of research was never looked back. From the tyrosinase, we went to the melanin. From melanin, we went to the biosurfactant. And that is the success story of our research laboratory. Now, quickly I'll be running through the slides. I'm not going through the slides. I'll be running through the slides because I'm aware of the time. How much time I left? Another 5 minutes. I'll have to run, right? That's how, please remember, all the isolates, we screen them, use the varieties of the different media, try to put it. If it is a brown color, then it seems that it is only the tyrosinase enzyme which is going to be produced by that particular strain. Intensity of the color has been equated with the darkness of the brown color. Lighter the brownness, then less amount of the tyrosinase. Highest of the darkness of the brown color, then more amount of the tyrosinase. So we have all these publications also. Next slide, please. And then straight away we wanted to standardize for the productions, bioprocess, other fermentation conditions. You can imagine, take the organism, standardize, optimize all the conditions. It's not enough if you do it in the 100 ml or 1000 ml in a laboratory scale. You have to update it with the advanced conditions, so-called as RSM, Response Surface Methodology, using a software tool. Next slide, please. That was standardized using the production of the tyrosinase, where we have the culture, we have the process variables which have been standardized. You can imagine 286.19 international units of the enzyme used to be produced. When it is subjected for this RSM technology, the achievement was 369.41 IU. That is the kind of the potential this strain had for the production of the extracellular tyrosinase enzyme, which was published in the Indian Journal of Experimental Biology, a CSIR journal. Next, please. Yeah, we wanted to show that. What is its application? I'm just telling, we have hundreds of applications by the tyrosinase. One such important application, what I'm trying to tell you is, based on the redox potential of this particular tyrosinase enzyme, we wanted to explore it for the detection of the varieties of the phenols in the natural samples, polluted waters, polluted soils. You know how polluted, if these conditions becomes increase of the different types of the phenolic contents. We know what is the destroying nature of the phenol content. Using this tyrosinase, very easily, one can detect the phenol contents present in the natural water samples and the soil samples. That too, we wanted to try with the composites of these nanoparticles along with the tyrosinase. It is something like a filter paper, small piece of the pH paper, dip it with this particular conjugate of the tyrosinase and a nanoparticle, dip it in the polluted water sample, and it will immediately turn to the brown color. Based on the intensity of the brown color, you can just say what is the amount of the phenol pollutant that is present in the samples. There are the best publications we had with this application point of view. Next, please. Screening of streptomyces for melanin. Yes, brown color. All these years research was focusing only from the enzyme involved in the formation of this brown color, and that brown color is nothing but skin pigment, a brown pigment. If you look into the understanding is melanin, we have a varieties of melanin. A natural melanin, which is a skin pigment of all the human beings. And we are surprised. We are more interested in the melanin than the enzyme. We have just moved a different directions of understanding the melanin. We are screening these organisms for the production of melanin. Next, please. Production of melanin, please try to remember this. We could achieve 5.29 grams per liter of the melanin. It's not an easy task. Melanin is highly insoluble in water. Highly insoluble in water. Whether the skin pigment can dissolve when you put the water? Absolutely not. That is a natural pigment which is insoluble in water. But the production was very rare. We need to sacrifice all the organisms to get the melanin natural pigment. Now we are really shocked to see that such an amount of potential this organism has got for the production of extracellular natural brown pigment. We explode it. Next slide, please. And accidental observations. Again, this is a fact in anybody microbiologist laboratory. See, we have the habit of not washing the utensils, washing the glasswares, washing the petri plates, washing the test tubes often in the laboratory. When we do something, we put it, we throw it in the sink, we go out. Remember, this is a similar incident that has happened in our laboratory. A student who is working on this melanin has left all the slants, which are supposed to be discarded in the autoclave, has dumped them in the sink, went to the holidays, came back after almost 1.5 months. And naturally, after having these observations for after 1.5 months, the kind of the droplets that were present on the surface of the slant of those cultures which are supposed to be discarded. And we were surprised. Can you see this? It's a slant culture. And can you see this dark gummy-like droplets on the surface of the culture? That was a surprise observation. It was just accidental observation. We never knew that this organism will produce such a droplet, and we never knew that those droplets are going to be what. Anyway, because of these important observations, we could take out those supposed to be discarded cultures back into the working table, start looking or analyzing that droplet. Droplet proved to be most important, purest form of the melanin. It is not just water insoluble melanin. You'll be surprised to say that droplet which has given us a patent, water soluble melanin, which was the first time report after 13 reports in the entire world. Ours was the 13th report to say that Streptomyces could produce water droplet, which is a water soluble melanin, which has got a tremendous revenue in the cosmeceutical industries. Any kind of cosmetics that is going to be prepared, normally used to be prepared using the melanin. It is difficult to convert the insoluble melanin to the soluble melanin, and the soluble melanin, if it is going to be explored in the industries, could get us a lot of revenue. Next slide, please. There is a history. We have patented. I'm not going into the details of it. You can refer that patent which has been already published. We could get the report as a patent. Next, please. Obviously, he was the student, and we had a lot of information, publicity in the press and media, and I put only the Hindu, that the breakthrough in melanin production that has given us a different path for varieties of the collaborations with the industries. That's how microbial technologies can take the sustainable future. Because of this, we had two major collaborations with the CSR laboratories, so-called as IICT, Indian Institute of Chemical Technology, Hyderabad, and also NCL Pune. That's how a whole this microbial diversity and their bioactive molecules led into the technologies for the welfare of the mankind. Quickly now I will be just running through the slides. Next, please. It's a simple characterization of the soluble melanin. You can look into the paper which has been already published. Next, please. Again, we could, one of the students who is a scientist in the CSR laboratory now, she could isolate the melanin from the Pseudomonas, which is much more superior than this Streptomyces, led into the different applications. Next slide, please. You can look, that is the kind of the applications. Melanin, Ray-Ban glass, brown color glass. Can you imagine? Please remember, there is a thin coat of the melanin itself. That's one of the applications. UV protectant. all ultraviolet radiations are going to be protected. Surface protecting agent, varieties of applications out of this melanin, particularly from the soluble melanin. Next, please. And please remember, we had a very good publications where we have proved that how this melanin could be used as sun protecting factor, which prevents sun-induced cancer. We have beautiful publications. Those who are interested, you can refer. And Jaishankar, I'll be leaving these slides with you. You can share with all the students. This is quite open. There is a indication from the esteemed chairperson that my time is over. Yes, that's how it has been proved. It's a flow cytometry, quantified it to prove that how it is anti-cancer best agent at a particular concentration. Next, please. There's a paper, Journal of Photochemistry and Photobiology with an impact factor of 6, which has been published very recently. A joint publication from our lab. Next, please. Melanin coated fabrics. Any kind of hair dye also can be used. Now it's a beautiful fashion, particularly among the girls and the women, getting the varieties of the hair dyes, particularly light yellow, dark yellow, brown color. So we are trying to make this project using the soluble melanin as hair dye and also fabric dye. Those who are interested, you can take this further because we are not progressing further in this particular area of research. Next, please. Biosurfactant. I'll stop at this stage because Neelam, are you here? Yes, Dr. Neelam Mishra is here, who is a DBT scientist. She has got a project on this rhamnolipid called as a biosurfactant. She is working on it. Because of the time, I don't explain all these slides. You can interact with her. She's here. She's going to present a paper on it, and you can interact with her for further details. Next, please. Next. 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 That is our collaborations with IIT, TCSR institutions. Next. So he is Dr. Syed Asghar, who is heading the NCM division NCL Pune right now. Please remember, scientist who is coming from the rural background, today he has got 47 new isolates, novel isolates, strains and the genera to his credit. You can just imagine what a number, the command this microbiologist has got over these novel variants. He is the man behind detection of all variants in the coronavirus. That is the kind of a command he has got on distinguished characters of all the microorganisms in general, viruses in particular, at the present scenario. Next, please. We have a bit of, he is Ranjun Lee. You can see that a father of actinobacteria. You can visit website, you can get all these addresses. Next, please. Next. Thanks to all the funding agencies, Government of India and Government of Karnataka for sponsoring our research project. Next. You can see this, a Betam Charla team of my research students, where we used to visit the field, collect the samples, get back. It's not an easy task. Those who are interested to spend at least 6 years in your lifespan, then only can do some kind of such breakthrough research. One has to be very patient to receive the again scolding from the guides, get yourself moving on to the breakthrough research. Thank you all.
Hello. 1 or 2 questions, because today is the keynote speaker requested us to interact. 1 or 2 questions. Anyhow, Professor Dayan Daksar will be with us. You can also interact with during the lunch time. Any clarification would you like to?
Which seems to be otherwise highly potential. If you are interested in the Western Ghats or this natural ecosystem, you can look for the exploration of the actinobacteria, which would be highly potential in serving the mankind through its varieties of the bioactive molecules. That's my submission.
So if no more questions, To summarize the keynote address, I've been lectured by Professor Dr. Dayanand Axel, the illustrious Vice Chancellor from Gulbarga University, Karnataka. I think he has given 45 minutes the lecture keynote address on the integrated diversity, bioactive molecules and technology for human welfare. He starts with the biodiversity aspects of the microorganisms with reference to the megadiversity nations, hotspots available. In that, what is the role of the microorganisms in the world, the evolution, and how its genesis is mentioned. And more about that, Professor has mentioned about the habitat for the microorganisms, especially he has screened in the limestone area, and he has mentioned about the role of the habitat and the role of the microorganisms in the ecosystem and then also in the niche, everything. And he has also screened the unique group of prokaryotic microorganisms with particular reference to the actinomycetes, and also has screened the samplings in quarries of the limestones, and thereby he processed many microorganisms that are very active in the media using the culture conditions and all that, and he made beautiful colonies in the limestone area where the habitat of microorganisms have been processed all together, and later he has been systemized with reference to the biosystematic studies from the basic, then molecular, and then also the other the conditions he has mentioned, and altogether he has isolated the novel, the microorganisms, which are new groups, is mentioned. Among the three important species he has mentioned, one is the Streptomycetes, the Gulbarga genesis, that was the most important. And not only that, he has characterized all the polymorphic changes of the microorganisms, bacterial system, and then potential applications also he has screened. And more about that, he has mentioned about the tyrosine synthesis, how it been produced and how it been bioprocessed, the molecule approaches, all he has mentioned. And very interestingly, it was amazing. I would also comment and also compliment to Professor Dayanand sir for production of the melanin. The melanin synthesis from artificially, and then also he has characterized the melanin, both are the biosurfactants and all that. So I think he has published many papers, and more than with the applause to him, he has patent. He has a patent in the melanin synthesis, and he's a high credit. Even yesterday also I mentioned in the CSA Institute that the production of whatever you are in your laboratory should be focused to the scientific arena. That is only route through your patent. So patent is very important. That I think he is the one who is a role model for the production of the patents from the melanin synthesis, and it is a great boon to the microbiologists who are coming to work in this area. And finally, he has mentioned about the role of the biosurfactants. I think he has given the overview of what projects have been carried out in his laboratory and with his students, collaborators and so on. I think definitely this area will be very helpful to the Indian microbiology the area, and not only that, and also for the international arena. So with this short note to conclude, my great heartfelt appreciation goes to Professor Dr. Dayanand sir. Thank you, sir. Thank you for your excellent presentation, sir.
Thank
Very
you, sir.
excellent.
With the submission of the stage, let me thank all my research students first for their contributions. Thanks to the organizers. Special thanks to my wife who has tolerated me all these 21 years without any botheration from the house. Because of her tolerance, I could achieve something here. Thank you once again to the organizer. Thank you, sir.
Thank you, sir, for your informative talk. As a token of appreciation for Professor Dayanand Aksar, sir, for his time and valuable contribution, may I call upon Professor M. Jaishankar, sir, to kindly present a memento to the lead speaker. I request all the oral presenters I call upon Ms. Anne Maria to introduce our lead speaker, Dr. Jaikumaran Nair, sir. I welcome Dr. Jaikumaran Nair, sir, on the dais.
A very warm good afternoon to all gathered here. In this prestigious occasion, I am standing in front of you with immense pleasure to introduce and welcome the lead speaker, Dr. A. Jaikumaran Nair, sir. Dr. A. Jaikumaran Nair, sir, obtained his PhD in biochemistry from the University of Kerala in 1993.
I welcome Dr. Prakash H. Halami, sir, to chair the session. And I welcome Dr. I.K. Manjula, ma'am, to be the rapporteur of the session.
A very warm good afternoon to all gathered here. In this prestigious occasion, I am standing in front of you with immense pleasure to introduce and welcome the lead speaker, Dr. A. Jaikumaran Nair, sir. Dr. A. Jaikumaran Nair, sir, obtained his PhD in biochemistry from the University of Kerala in 1991. He has worked for his PhD in the National Institute of Interdisciplinary Science and Technology, formerly Regional Research Laboratory, Thiruvananthapuram, Kerala. After the submission of his thesis, he has worked as a fellow scientist in National Institute of Interdisciplinary Science and Technology. After receiving PhD, he joined the Indian Agricultural Research Institute towards the end of 1992 as a research associate. He received Young Scientist Award for his work carried out in the Indian Agricultural Research Institute in the year 1995 in the Kerala Science Congress. He has joined the Center for Genomics and Integrative Biology in 1995 as a CSIR Research Associate and later joined the Center for Biotechnology, Jawaharlal Nehru University, New Delhi in the same year. He received Young Scientist Award as a project by DST in the year 1999, and he worked as the principal investigator of the project in the Center for Biotechnology, Jawaharlal Nehru University. In 2001, he joined Bundelkhand University, Jhansi as a lecturer and later joined MIT University of Biotechnology as associate professor in the same year. He joined the Department of Biotechnology, University of Kerala in 2003. Dr. A. Jaykumaran Nair sir has been working in the field of microbial enzymes, therapeutic compounds and proteins, monoclonal and polyclonal antibodies, recombinant vaccines and proteins for the last 22 years, including the tenure in University of Kerala. His interests also include characterization of new proteins and genes, protein engineering, development of bioprocess technology for the industrial production of microbial enzymes and products. At present, he is working on various types of microbial enzymes, therapeutic proteins, and in the development of ELISA-based diagnostic kits for cancer and metabolic errors-related diseases and recombinant vaccines against poultry and cattle. His laboratory is also engaged in the screening of bioactive compounds from the natural sources, its identification and structural characterization. And I am proud to introduce and heartily welcome you, sir. We hope your presence may make a change in the young minds. Thank you.
Thank you, Anne. I request Dr. Jaykumaran Nair sir to deliver a talk on the topic Microbial Biotechnology, the perfect blend for sustainable development.
Good afternoon, respected Dayan sir, Ellen sir, or Jayasankar, and other dignitaries and my friends and students. So today's talk is about the sustainable development, how the sustainable development can be promoted by the involvement of biotechnology, particularly microbial biotechnology. We are talking about the development. As you know, development is very essential component of our society as well as the nation, and any development that of course result in pollution, and thereby because of the pollution it will result in the degradation of environment and ecology, and finally it will result in the loss of lot of biological resources. So without any pollution, we cannot do any development. But how we can bring down the intensity of the pollution, and how we can solve the pollution that already created by the development. So that is what the thermodynamics also states, that means anything that happens, whether it is a physical process, chemical process, or any other type of development that results in the formation of some disintegration in somewhere, in some part of the universe. So that is what the second law of thermodynamics correctly expresses, and we know that it is very much applicable in the case of biological systems, because we know any development, as far as the development of a cell into a complete organism, essentially it appears to be a development where there is no disintegration. But as in the case of photosynthesis, then also we can see that carbon dioxide and water that comes together and forms the carbohydrate. But we cannot see any disintegration anywhere in that nearby things. But of course, when we are see how it gets the energy for that processes and which is the source of that energy, then we can see that the place where the disintegration occurs, that we know it is in the formation of the light and the heat from the sun because of the fission and fusion processes. So anyway, here the involvement of biotechnology, how we can reduce the intensity of pollution and how we can replace certain type of industries with the biotechnology industries. We have seen that what is sustainable development. So development means it may not keep the view that we have to or we see the future also. But we know in the history also we see that there are a lot of developments by which the disintegration has occurred in the environment. For example, I will give an example from the World War I. So there they want explosives. So explosives, they used acetone as one of the ingredients for the development of for the explosives. But the acetone, they synthesized or they extracted from wood. So they wasted or they destroyed a lot of forest for getting the acetone, and thus there was deforestation in the areas of England and other parts. Thus the acetone production was stopped. And at the time, a microbiologist, a Jew microbiologist from Israel, means they were in the all over the world, Jews were there. One fellow among that discovered a bacterial strain which can produce acetone from starch. So immediately, the Prime Minister at the time of, I think, at the time, the British Prime Minister, he asked that person, scientist, to develop a bioprocess. I think that was the first bioprocess, bioprocess for sustainable development, the first example for that. He developed a bioprocess for the production of acetone from starch, and this means he developed that process all over the world, including India, in Gujarat. And that person was the wise man. And he was the first president of Israel also, because after the First and Second World War, the countries, they given their land back to them because of this particular service that has been given, that is the development of this microbial. So I given this example as a first example for the sustainable development, means by keeping without destroying the environment, they developed these things. At the same time, we know that the other effect of that process, but acetone is very important product, stock, chemical for the industries now. And the sustainable development, as you see, it has to keep the future also. So the development, that today's development should not destroy the future and the future generation. So keeping that mind, in our mind, we have to do the development, and that is what is called the sustainable development. And biotechnology can contribute a lot in this sustainable development. So we know the normal industries, or we know the chemical industries as well as the other industries, where it results a large amount of pollution, and that with different type of pollutions, you know,
what are the type of pollutants that is created by the industries. Air pollution is there, land pollution, and so many type of pollutions are there. All these will create different type of effects in the ecology as well as the environment, and result in the even the loss of certain species of certain means it disturb the biodiversity also, resulting in the loss of larger number of species. So here in the sustainable thing, biotechnology can replace certain type of industries by green technologies. So without creating pollution or by minimizing the pollution, biotechnology can contribute certain green technologies. That is the things that we are looking for. And before that, everybody knows what is biotechnology and that, the definitions of that. But only thing is, if you ask what is meant by biotechnology to different person, their approach, their definition will be in different form. But anyway, there is the involvement of biological systems in that. And biotechnology can give clean environment. Means it can, whatever pollution is there or pollution that is created by even by the biotechnological industry, it can be cleaned by the biotechnology, particularly microbial biotechnology. So it involves in the conservation of biodiversity. So there are so many methods by which we can conserve not only the plant biodiversity, animal biodiversity, but even the microbial biodiversity can be conserved and protected by the biotechnology. So we have seen in biotechnology there are large number of branches. Each branch involves the activities in that particular area, contributing to the different developments in that particular area. One of the key aspect of biotechnology is microbial biotechnology. About 60 to 80% of activities, development or researchers is in microbial biotechnology. But of course, there are other branches which contribute to the developments in different areas like plant biotechnology, animal biotechnology, agricultural biotechnology, environmental biotechnology, nano biotechnology, and bioinformatics and computational biology. So like that, so many branches are there. Still now, there are new branches developing in the field of biotechnology. So this is the microbial biotechnology, how the microbial biotechnology can contribute to the sustainable development of the nation. So each branch, it has got different aspects and different products. So generally we call it as microbial products. But even though it is called microbial products, and also processes are there, microbial products as well as process. So in the biotechnology, there are three aspects that is applicable in the case of microbial biotechnology also. Of that three components, that is the first one is biotechnology should produce a specific compound like ethanol, antibiotics. So the compound should be very specific and it won't be a mixture. Okay, so that is the first part of the biotechnology. Second one is the process. Processing means it converts one material into another form. The best example that we have is the formation of curd, the processing of leather, etc. There is no specific compound, but a material is converting into another material. So that is the second part. And the third one is the formation of transgenic organisms. So transgenic organism is also very old system, means the process, but there that is by hybridization. So by hybridizing two organisms, certain characters will be transferred to the other, but that is not very specific. But now by genetic engineering, we can take a particular DNA or a gene and transfer into any microorganism or any other organism. So there won't be any barrier for the transfer of genes. So there is the transgenic organism, the creation of transgenic organisms. So all these are there, and product, process, and the transgenic organisms. Here, the environmental applications So we're going to talk one by one. So the first product that is very essential and having a lot of applications is microbial enzymes. So microbial enzymes, so that is available in other organisms, also in plants as well as animals. But the advantage of microbial enzyme is it is very easy to obtain, and by the process of bioprocess technology, the quantity of enzyme can be enhanced by applying different type of things. And also there is the process of strain improvement, thereby new type of organisms which can produce large amount of active enzymes can be achieved. So these are some of the enzymes that usually get from the microbial systems for different activities, including the food processing, like starch industry, the production of ethanol, and so many other essential compounds like various types of acids, vitamins. So many compounds are there, even compounds like cyclodextrins, bioplastics, etc. Then the medicine, so some of the compounds among that, the enzymes that listed above, that includes medicinal compounds also. So medicinal compounds means the compounds which we use for medicines like antibiotics. Then there are other secondary metabolites, then certain therapeutic enzymes. For example, the enzyme that is fibrinolytic enzymes. So one of the very good example for the therapeutic protein or therapeutic enzyme is fibrinolytic enzymes. That also we will discuss little bit these details. Then the vaccines, for example, pathogenic bacteria, if there is, we can use that pathogenic bacteria for getting the vaccines by cell culture systems, then vitamins, amino acids, and so many other veterinary medicines. For example, the vaccines of chicken as well as other cattle, etc. So the food and dairy products. One of the major application of microbial biotechnology is food biotechnology as well as dairy biotechnology. So we know that large amount of or large number of dairy products are available now, including the milk products, curd and related products. Then the processing of food. So there are large number of processed foods, processed starch, processed protein as well as lipids, they are available in the market. And then the organic acids, glucose, fructose, and other mono and oligosaccharides. We know that glucose and fructose syrup is very important in the pharmaceutical industry for the production of certain medicines as syrups. We know the fructose is more means it's sweetener than the glucose, so that is preferred for the pharmaceutical industry for making this type of syrup. Similarly, in the other industries, for example, the bakery industry or making toffees, there we know that we have certain preparations where the interior part of that particular material will be liquid or semi-liquid. For example, biscuits and such type of toffees are internal component is liquid and with the solid core outside. And biotechnology has got some important role in that. For example, so in the internal part will be sucrose. Normally they fill with the sucrose along with the certain enzyme, invertase enzyme. And in course of time during its preparation, the invertase will act on that sucrose and convert it into glucose and fructose. And this fructose and glucose both, since it is more hygroscopic, it will take water from the around the materials and slowly it becomes liquid. So thus we get an interior part which is semi-solid or as liquid. So the biotechnology has got some role in that area also. Then we know the other microbial compounds, and the best example is cyclodextrins and etagonic acid for acrylic resins. Similarly, there is other biopolymers, polyhydroxybutyrate. It's one of the major materials that is used for biopolymers or biodegradable polymers which can be replaced. these plastics. So it's called bioplastics also. Similarly, lactic acid. Lactic acid is another material which can be polymerized and converted into biopolymers or bioplastics. But of course, it's not like the ordinary plastic. There will be, the use will be limited because it will be biodegradable. But of course, we can promote that and thereby we can extend the lifespan also. But when we want to degrade it, it will be degradable, unlike the other ordinary plastics. Then the biofuels. Biofuels, there is biofuel cells. Using certain microorganisms, we can make cells to generate electricity, and thereby we can make even batteries of that, and thus electricity can be produced from microorganisms by certain specific adjustments. So that is a coming up research area where a lot of purposes are coming up. Then, of course, the bioethanol. Bioethanol, ordinary ethanol is the same thing, so but it is designated as bioethanol because of its special uses. So it has to be very clear if it is used in the pharmaceutical industry. Similarly, this ethanol can be used for making biodiesels also by mixing with the other type of the ordinary fuels, the fossil fuels. So it is a very important area where new type of fuels, biological fuels, can be achieved, and that can be used for the replacing the ordinary fuels, that is the fossil fuels. Then environmental applications, as we have discussed earlier, one of the major application of so it's called environmental biotechnology. So there, the waste management and detoxification of materials. So waste management is one of the major area of environmental biotechnology, as well as the ordinary in the means any city or anywhere a large amount of waste is means created, and as we have discussed, that it will be a part of our development because waste has to be managed. Waste will be forming as a part of the development, and it has to be properly managed. So waste is only a term that we are using. If we are finding a use for that, it won't be a waste. So that is the attitude that we are now developing. So nothing is waste if we are finding a use for that, so that all the waste has to be reused, or it has to be value added and converted into useful forms so that it can be renewed, and the development will be sustainable. Then the effluent treatment. Effluent treatment, we know large amount of effluents are generated from the industries, like a titanium. Large amount of effluents that is coming out of the industries, and the effluents will always have some toxic components, and that toxicity can be either reduced, or it can be converted, or it can be detoxified by the use of a large amount of, or a consortium of microorganisms. So that is one of the major activity in the environmental biotechnology, and that also is very important in the sustainable development. Then, as you know, the SCP and SCL, so they are the byproducts. It means, as I told, the effluents, for example, the effluents of starch factory. So starch factory, that will have a large amount of organic load, and this organic load can be converted into single cell protein as well as single cell lipid, and it will be very useful for making the food products, so different type of food products. And also depending on the effluent, what type of effluent you are using, the SCP or ACL that is generated can be an ingredient or supplement to the foods, or it can be converted into cattle feeds as well as poultry feeds. Then the agrochemicals like biopesticides, bioherbicides, and genetically engineered organisms that also can be used as biopesticides for protecting the agricultural plants, and also the nematicides. not So now I'll just give some examples from our laboratory which we have conducted. First one is we have a work on cyclodextrin and the CGTase production. Then the fibrinolytic enzyme from bacteria, that is Bacillus cereus. Then alginate layers, Nocardia species, endoglucanase. Endoglucanase means it's a cellulase, component of cellulase from Aspergillus. Actinomycin D that we know, but we have developed a process by which the production, the quantity of actinomycin D can be enhanced. Then alkaline metalloproteins, alkaline lipase, and lactase from Arthrographis and Enterobacter cloaceae. So these will one by one very briefly. So the first one, cyclone. Before this, there is another one also. That is the recalcitrant chitin. Chitin, you know, the powder or the husk powder that has been created in the kaya industries, and that will be very means it cannot be biodegraded, because it is recalcitrant and it will take a long time. So we developed a process by which these huge heaps of chitin can be converted into manure, nitrogen-rich manure by using certain microorganisms. That is the nitrogen-rich compost from coconut pit, and that we have conducted in association with the Kaya board. And it was very successful, and it has been transferred to the needy persons also. The other one, the cyclodextrins. So cyclodextrins will come to the next slide now. And sorry, the previous. We have isolated keratinase. Keratinase is another enzyme which degrades the hair and as well as similar type of recalcitrant proteins, insoluble proteins like nail, hair, etc. So these are also very important because we know that a large amount of waste, the poultry waste or chicken waste, is generated in certain areas, and people usually go dump it there, but that can be used and using this keratinase or keratin producing microorganisms can be used for degrading these recalcitrant solid proteins, and means like a chicken feather, all these are there. So it can be converted into certain value added products and can be converted into poultry or the cattle feed or some other compounds by which the pollution and these things can be, these waste can be managed properly. Next one. So these are some of the other type of researchers that already have in our laboratory. One of that is alginate layers. So alginate layers is another therapeutic protein. You know, alginate layers is a protein or enzyme that converts calcium alginate or any type of alginate, and it breaks into certain type of monosaccharides. And it is a therapeutic protein because it is the medicine for a type of what is called the lungs problem, and it's a genetical problem. Because of the genetical means the defective activity of some genes, there will be the exudation of calcium ions into the lumen of lungs. Along with that, this calcium forms a platform for the formation of infection by pseudomonas and other type of infective organisms, and that it will block the and this calcium and this microorganism, this pseudomonas produces alginate, and this alginic acid. this alginic acid and this calcium together form calcium alginate, and this calcium alginate is fibrous, and it can very well block the lumen, means the cavities of the lungs, and thus it can block the air passage. So it result the what is called a type of asthma that result in the death of the person. So this calcium alginate layers is a very good medicine for that, and by spraying this calcium alginate layers to the lungs, it will degrade the calcium alginate that is formed in there, and it will make the air passage little bit clean so that the breathing will be easy. So not only that, along with the calcium alginate, this infective organism that is already there, they will die because of the use of antibiotics, and the DNA that from that organism, along with the calcium alginate, form little more fibrous materials by which the lumen of the lungs will be blocked. And this can be rectified or can be cured by using a mixture of alginate layers and DNA together and spray into the lungs through mouth or through nose. And so that is the importance of alginate layers. Then, as we know, the keratinize. Keratinize has got some cosmetic application also. For example, we know there are some hair remover materials that is available in the market, and this hair remover can incorporate this keratinize, so that the removal of hair will be very easy and it will be better than the other one. Then the agnomation I have discussed, then the facial cream. So that is not related to microbiology, but of course we have developed a honeybee-based facial cream with honey bee gel. So that is another product developed in our laboratory. Now we can come to the next one. So this is about the CD cyclodextrins that we have developed in our laboratory, and we have isolated a number of organisms. One of the organisms is this one Leucobacter species, and its species were not identified. You know, the cyclodextrin is an oligosaccharide. Ring means a cyclic oligosaccharide, in which 6 to 8 glucose units can be joined together and form a cyclic compound. And the peculiarity of this compound is that the hydroxyl group of this assembly will be protruded outside. So it will be easily soluble in water, but the interior part, the cavity part of the cyclodextrin will be hydrophobic. So that the advantage is it can take hydrophobic compounds into that lumen, and thereby it can act as a host-guest interaction. So if you want to dissolve some of the insoluble materials, insoluble compounds in water, you take it in cyclodextrin and dissolve it, so that you will get a solution with that particular compound, and with the principle of what is called the process of based on the concentration, the compound that is present in the lumen of the cyclodextrin will be coming out slowly, so that the slow release process that also is applicable, so that it has got tremendous application in pharmaceutical industry, in food industry, as well as in other various types of industries where these guest host interaction is needed. For example, in the case of pharmaceutical industry, the slow release, that is encapsulation, a type of encapsulation is needed. Thereby, the medicinal compound will not be released at a stretch, but slowly it will be released based on the concentration factor. So when the concentration becomes equal, then we have to take another dose of that compound. So that is the importance of cyclodextrins, and the cyclodextrins has got a lot of applications, and the cost is very high, so that we are trying to get more of these organisms which can produce this type of either the cyclodextrins or its enzyme, that is what is called the gluconotransferase, cyclodextrin gluconotransferase, CGTase, either the enzyme or the organism, that will be better. And also we have another activity there. We are planning to take this gene of cyclodextrin gluconotransferase and transfer into tapioca plants with a promoter which is very specific to the tuber. so that we believe that we can get the enzyme expressed in tuber itself, and this enzyme in course of time convert some component of some part of the starch into cyclodextrins, and so that we can directly get the cyclodextrin from the tuber very easily. So if it is achievable, means it can be, if it can be achieved, it will be a very important outcome. Next one. So the next one is about the fibrinolytic enzymes. So the fibrinolytic enzyme, there is also a therapeutic protein which is used, which we have already the fibrinolytic, these clot digesting enzymes, which we used for removing the clot that is formed in the heart or in the circulatory system, either in the heart or in the brain, to prevent the hemorrhage as well as heart attack. When there is a block, we use this material for releasing that particular block. Now, whatever we have is having some side effects because it is protein digesting enzyme. It's not very specific, but we have isolated fibrinolytic enzyme, which is very specific in the sense it directly attacks the fibrin that is formed, or which is the main component of that block, and thereby it reduces or it dissolves that fibrin so that the block is removed. And that compound, that enzyme, that is it is generally called a fibrinolytic enzyme, and we have isolated the bacteria from soil, and it shows this activity so that we cultured that bacteria and isolated the enzyme and purified it, and also we characterized it. So these are some of the experiments or the research outcome that has been carried out in our department, and we show this, or I expresses this here as an example for the involvement of microbial biotechnology in the sustainable development, or the development without creating any problem for the future. So with that small talk, I like to stop it here, and I am very thankful to all the audience here for patiently hearing this. And I express my gratitude to Dr. Dayan Jaishankar, as well as Sri and other Manjula for giving this opportunity. And also I am thankful to Dr. The president of our MBSI Microbial Society. So he only suggested my name to Dr. Jaishankar, and I am very much thankful to him also for giving this opportunity. So thank you very much.
Thank you, Dr. Jayakumar, for your wonderful lecture. This is the first lead lecture of this today's conference, and Dr. Jayakumar has very elaborately explained how the microbial biotechnology can really work on the sustainable development. And everyone knows that sustainable development goal of United Nations organizations, which also emphasizes the role of biotechnology in a bigger way. And definitely the sir, which he has covered the several areas with reference to the conservation of biodiversity, whether it is plants or microbes or the animals, as well as the different microbial products, which was very important, and which most of the researchers also are taking up, that includes the microbial enzymes, medicinal compounds, food and dairy products, biopolymerals, biofuels, and several other environmental application and bioproducts also. And remaining part of his lecture, he has actually emphasized the R&D activities which is being carried out in Kerala University. So it was actually very wonderful to learn and listen from your side. We know that I think time is a little bit short, but still we should have one or two questions from the audience. One from that side and one question from this side. Any questions, please, from the audience? Rather than they asking among themselves, they have to ask here to the speaker.
Otherwise, I am going to ask them afternoon session. No? All they are waiting for lunch, I believe.
Okay.
So I take once again this opportunity to thank on the behalf of organizers to the speaker. I thank the organizers to give me this opportunity on behalf of my reporter also for giving us this session to lead. Thank you very much. Over to the organizers now.
Thank you very much, sir, for an interesting talk. As a token of our appreciation for Dr. Jaykumaran Nair, sir, for his time and contribution, I call upon Dr. A.K. Vinu, ma'am, to kindly present a memento to the lead speaker. I request all the oral presenters to kindly submit your PPT slides to the reception counter in the lunch break. Yeah, to the technical committee. And I request all the poster presenters to kindly kindly keep your posters over there. It has been the slots has been given, and the slots has been given in your kit as well. And I request everyone to gather at the quadrangle for the lunch and kindly assemble in the auditorium for the upcoming lead talks.
there is an announcement. Those who are presenting poster as well as oral for today's session, you have to submit your oral presentation PPT at the registration counter, and those who are presenting posters, they have to display their posters at the corridor according to your token numbers. Those who are doing poster presentation, please display your posters according to your token numbers. ಪ್ರಾಣನ ಅದು ಬಿಟ್ಟರೆ ಯಾವ್ದು ಬರಲ್ಲ.
Hello. 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 Hello.
Good afternoon, everyone. I welcome Dr. Jaykumaran Nair, sir, to be the chair of the lead talk session, and then I welcome Dr. Geetanjali ma'am to be the rapporteur of the session. Now I call upon Ms. Kriti HK to introduce our lead speaker, Dr. Parli Venkateshwara Bhaskar, sir, who will join us online.
Good afternoon, one and all present here. I'm Kriti from First MSc Microbiology. I'm extremely privileged to introduce and welcome today's lead speaker, Dr. Parli V. Bhaskar, Scientist E, NCPOR, Goa. He pursued his MSc in Microbiology from Bhavnagar University in 1994. He received his doctorate in Microbiology from Goa University in 2004. He started his research career in 1995 at CSIR NIO as project assistant and thereafter as a CSIR SRF, completed his doctoral thesis on microbial EPS and their role in marine food web and carbon flux. A part of this research work was carried out in Germany as a UGC DAAD research fellow. As a research associate at CSIR NIO from 2003 to 2006 under SIBER project, he studied the roles of microbes in DMS production in coastal water and isolated phototropic bacteria from hypoxic coastal waters of Goa. He was a GSPS fellow from 2006 to 2008 at Nagoya University, Japan. He worked as an invited postdoc researcher at University of Algarve, Faro, Portugal from 2009 to 2010. He joined NCPOR in 2010 and since then he focused on microbial diversity, microbial activities, DOM, chemistry and distribution of microbial siderophores in polar waters. He was a member of SOOS SLC from 2012 to 2017 and is currently the national representative for India at SOOS. He was also a visiting lecturer at University of Algarve at Faro, Portugal for Erasmus Mundus Program 2015. He is also the associate editor for BMC Microbiology. He has 35 scientific publications in peer-reviewed journals, 2 book chapters, 3 technical reports, 30 papers, abstracts, posters in symposium and conferences. He has participated in numerous expeditions in Arabian Sea, Bay of Bengal, Gulf of Cadiz, Tokyo Bay, Indian sector of Southern Ocean, and the Arctic. Thank you.
There is a short announcement for oral presentation. For today, number 1 to 11, those who are enrolled for oral presentation, from number 1 to 12, they have to give their PPT file at the registration counter. And for the poster presentation, token number 1 to 14, they have to display their posters at the allotted area.
I request Dr. Parli Venkateshwara Bhaskar, sir, to deliver a talk on the topic Polar Microbiology, Insights into Environmental Microbiology and Biotechnology.
Good afternoon. I hope I am audible to one and all. At the onset, my apologies for not being able to meet this conference because of inclement weather in Goa and en route to Bangalore. I'm thankful to Bangalore University and Professor Jaishankar Madaya for giving me this opportunity to be one of the lead speakers in the august presence of biotechnology heavyweights who have earlier spoken varied topics, including bacterial diversity, use of microbial technology, various solution of various compounds and biomolecules. Today, I'm going to introduce the audience to polar microbiology. So, what is so unique about polar microbiology and why it is so interesting? If I'm not visible, you may please let me know. If there is any issue with the slides also, you may please let me know. I hope I'm audible to one and all because I'm not getting any feedback from the other end. So I would like to know whether I'm audible clearly or not.
Yes, sir.
Thank you. So today I will introduce the audience, mostly the young researchers, about polar microbiology and those who are interested in carrying out biotechnology work or environmental microbiology work. I would give them an idea about what are the possibilities, what are the limitations, and what are the pitfalls. So on the screen you can see a typical scene of the polar environment. What you are seeing backdrop is the iceberg, and the vessel that you are seeing at the lower panel is has been our research vessel when we are going down to Antarctica, Antarctic coast, for carrying out marine sampling. Behind this vessel, you can see there is another vessel on the backdrop, which is the vessel which is carrying transshipment for the Indian Antarctic base, RTA, in the Antarctic Pits Bay region. So the entire presentation will be kind of divided into two major parts, but it won't be very just. I'll be explaining you about polar microbiology, various kind of works that have been done, what kind of opportunities in biotechnology, especially in goals. What are the limitations? What is the existing policy? And what are the challenges and opportunities that as a young researcher we would always like to know? Remember that when we are working in polar regions, it is an international area. It's outside India, so there are certain policies that we need to consider with such activity. There are some limitations which we cannot transgress, unlike what we can do at our backyard. Whether it is a terrestrial sample or a forest sample or a riverine sample, we know the backyard. We know the rules, but when we are going to Antarctic, we need to be a little bit more careful about the international rules that are applicable when it comes to carrying out research in these regions. So, a general introduction to polar microbiology. You know that we have different forms of microbiology: medical microbiology, terrestrial microbiology, food microbiology. So, polar microbiology is a very specific region. The planet talks about microbes which are living in the snow, ice, glacial lakes, glacial environment, and including the atmosphere in and around the poles. So the unique feature of this environment is that it is an extreme environment. We have extremely cold and dry conditions. 6 months, whether you are in the Arctic or whether you are in the Antarctic, 6 months it's dark, and 6 months we have light. In that also the peak summer season is hardly for a month or two maximum. Rest of the time the sun is just above the horizon and it sets down. Some time. So because of the depleted ozone in the atmosphere, in the poles, we have higher UV radiation in these regions. So accordingly, the flora and fauna have adapted to this environment. The temperature fluctuations, unlike in equatorial regions where you may find extremely hot conditions like 45 degrees in summer and extremely cold conditions like sub-zero conditions, but in the polar environment, the temperature fluctuations are always sub-zero. Even there, if you have a slight variation in above zero conditions, then it's considered to be a huge anomaly in the atmospheric temperatures. So at times in the Arctic, temperatures have shot up to 30 degrees. Yes, it's an anomaly, but it has happened. But the average temperatures in Arctic would be around -4 to 12 degrees and 0 to -20 in winter. And pardon? So yeah, so the microbes which are growing in this environment are also very unique. Their metabolic rates are slow, except in the summer when they get some amount of sunlight. There is melting of glaciers. There is an increase in nutrient supply, and the microbes start growing much better. It's a faster rate than normal conditions. So unlike other major extreme environments like deserts, hydrothermal vents, hot springs, volcanic mounts, which are hot in conditions, deep biosphere, or acid mines, which is having extreme pH condition. Polar ice caps and sea ice environments, which are extremely dry and extremely frigid environments. So let's have a quick look at the global microbiological activities which are being carried out under polar science. So earlier, the polar research used to be concentrated only in Arctic and Antarctic, but of late we have included mountain glaciers, which includes, that's why we can see here the pink spots over the Indian Himalayan region, Tibetan Plateau, also in the Arctic. You can see here the mountains on the west coast of USA, Alaska, and along the coast of South America. There are mountains which are having glacial lakes, and these are also equally important. So they also provide a polar-like environment because they are permanently frigid and sub-zero temperatures. Globally, the polar research is carried out or rather governed by SCAR, that is Scientific Committee on Antarctic Research. It's very specific Antarctic research. International Arctic Science Committee, which is very specific for Arctic region, and SOOS, that is Southern Ocean Observing Systems, which is also very specific for Southern Ocean. Now, in this map on the right side, you can see what we call as Southern Ocean is the ocean which is circulating around the Antarctic. And you can see there is no landmass, no land barrier here. So there is, interconnection of water masses between the Pacific, the Atlantic, and the Indian Ocean, and they get connected and they get mixed in this region. And this is a highly dynamic environment, strong seasonal variation in temperature. Have a geographical latitudinal variation in temperature. Also undergo the day, light and dark conditions very similar to that of Antarctic region. So this environment is very typical, although it's marine, but it's unlike what you find in the northern hemisphere. Even if you go to Arctic, the conditions are very different than Southern Ocean. So on this panel, you can see the red dotted line is an approximate demarcation of the Arctic Circle. And you can see Greenland, this is the Arctic region. And the difference between the upper panel and the lower panel, you can see that the upper panel, the North Pole is actually nothing but sea ice in the winter. And in summer, it is very thin sea ice nowadays because of the global warming. Whereas in the south, the southern South Pole is actually situated on a landmass of Antarctica, which is covered with ice. And then the sea ice extends all the way up to 55 degrees south. So these two are unique environments, as you can see. The Antarctic region is disconnected from any other landmass, so it's an island continent by itself. Whereas if you can see the Arctic is actually an ocean which is surrounded by landmass. So that's the unique difference that you find in this place. Because it is surrounded by landmass, there is more research being done in Arctic, especially from biotechnology point of view, because accessibility to the area is there. Different countries govern their area in their own way. So there is all kind of research, exploratory research, and biotechnology-based research being carried out in Arctic. In Antarctic, yes, they do carry out research to understand the biotechnologically important microbes. But you can see that it's disconnected from any other land, and Antarctic, as it is not owned by any country in the world. It is open for scientific research alone, and no commercial activity can be carried out in and around this landmass and the oceans surrounding it. So the other part of microbes that you can study in polar region are the permafrost. Permafrost are the terrestrial-based environment. It's a permanently frozen water, ice, which is subterranean. Now, normally in the tropics, you will find that the land holds water or moisture, but because it's a polar region, this is always frozen, and that's why it's called permafrost, permanently frozen. And in the recent years, with the increasing temperature in the poles, there is reports of increased melting of permafrost, thereby making the dormant bacteria getting active with the availability of water, sunlight, and slightly warmer temperatures. So a lot of research is being done to understand what lies underneath the permafrost, what kind of microbial diversity is there, what kind of microbes can be isolated for biotechnologically important compounds, and also pathogens. Any hidden pathogens in the permafrost, we are not sure what they have been, because they have been buried in this area for the last millions of years, and now they are getting slowly active. We don't know what kind of pathogens may escape from permafrost. So a lot of research is being done in the Arctic, especially to understand the permafrost microbiome. And this lower panel, you can see it's a sea ice microbial community. I have taken this from a cover photo of a journal, and you can see a beautiful mat of sea ice algae. Now, you may say what microbes have, what microbiology has to do with algae. Once we are in the area of environmental microbiology, we cannot restrict only to bacteria. In nature, nothing works alone. Bacteria don't grow in isolation. They don't grow in an insulated environment. They are always interacting with other microbial organisms, which includes microalgae like diatoms. There are other cyanobacteria. They are all growing together. And this CIs, what you are seeing is a very good example of what we also call biofilm. So it's a biofilm of microbes which is growing trapped in a sea ice. So it gets exposed during summer when the ice melts, and during the winter, whatever microbes are there on the surface water, they get trapped in the sea ice and they remain dormant, or they remain metabolically active for the winter, but they are inside the ice. So there is a huge community of diverse microbes living in the sea ice, which have potential to survive the winter, mainly because they produce compounds like exopolymers. They have compounds which doesn't allow them to freeze, antifreeze compounds. So a lot of things are there which can be explored in sea ice algae or sea ice microbial community, which is called SIMCO. And they also give us a vast opportunity to explore into new kind of biomolecules, which might be very endemic for this region. So polar microbiological research in India is normally in the area of Arctic, Southern Ocean, and Antarctic. Recently, we have also started Himalayan glaciers. So in NCPUR, under the Ministry of Earth Sciences, we are now focusing on Arctic region, Southern Ocean, and Antarctic region, and also Himalayan glaciers. This is the for the Arctic studies. We go to this place called Svalbard Archipelago, which is north of Norway, and then there we have been studying this Kongsfjorden and Krossfjorden system. Along with that, there have been people from University of Trichy and other BHU who have been isolating cyanobacteria from the terrestrial streams and ice, snow and ice meltwaters, the land surrounding these fjords, and also the sediment samples. There's a big number of research, a good number of papers have come out of Cochin University of Science and Technology, who are working on sediment microbes and the Southern Ocean, as I already mentioned, is the ocean region around the Antarctic. These are the regions internationally which has been identified as key areas where research needs to be done in order to understand the biodiversity of all flora and fauna, including microbes. So, as I mentioned earlier, Southern Ocean includes marine microbiology, sea ice microbiology, and sediment microbiology. And in Antarctica, we have biomicrobiology, lake microbiology, and pollution-related studies. So, okay. So here you can see that this is at the onset of an expedition to Antarctica. This is the same vessel which you see here in the lower panel. So it's just before leaving from Mauritius. Our expeditions, they start from Mauritius. We go all the way to the coast of Antarctica. We carry out sampling all the way by going and returning, and also along the coast of Antarctica. Our expeditions don't touch the land. We are not given the permission to land. Whereas in Arctic, which is on the right side of this panel, you can see on the upper panel, that's myself, Professor Hata from QSET, and he's from professor from University of Trichy. This is in the Kongsfjorden. You can see the glacial background over here and the mountain ranges. So this is the area which we carry out sampling. Samples were collected from sediment, water, and even landmass, permafrost samples. So before we think of carrying out biotechnological activity, researching goals, we need to understand the restrictions that are imposed to researchers. There is no restriction per se as far as research is concerned, but India is a signatory of the Antarctic Treaty, and as per the Article 3 of the Antarctic Treaty, in order to promote the international cooperation in scientific investigation in Antarctica, the contracting parties agree that to the greatest extent feasible and practicable, information regarding all scientific programs have to be made public to all the members who have signed the Antarctic Treaty. And this is to make sure that, unnecessary transport and unnecessary expenditure behind the expeditions don't take place. Scientific personnel who are landing on Antarctica, they have been exchanging, they need to be reported immediately, and any observation and results from Antarctica shall be exchanged and made freely available. So now what happens is, suppose you find a sponge, a marine sponge, very typically found endemic to Antarctica, and you find that, you can extract some really valuable compound from that. Well, that's the information you have, but you have no rights for any exploration. So no country has any right for any exploration of any living resources in Antarctica. It's a sweeping ban on all participating countries that they cannot use that land or the surroundings of that land for any commercially explorative activity. It is not allowed. So if you are looking for biotechnologically viable molecules, it's good that you get it, but the cultures you have to maintain under those conditions, which is found in polar region. And if you are unable to maintain it, bad luck for you, but you cannot think of just extracting the resources from there at a commercial level and saying that we found this compound, it is interesting, we would like to exploit it at industrial level. That's not possible. So any organism that comes out of Antarctica and is getting published, you cannot patent it, you cannot commercialize. Yes, you can commercialize the product, but you cannot commercialize the organisms, the living resources. However, bioprospecting in Antarctica has certain legal issues and limitations, especially those are looking from investment point of view for private companies, whether we can go there and do any kind of exploration for commercial purpose. It's a strict ban. There is no way anybody can do that. So this poses questions when it comes to patenting of materials and ownership information. It has to be shared. So that's why I'm saying that when we think of biotechnologically important molecules, everybody looks into the commercial aspect. That when we commercialize it, whom will we give the credit, or who will get the copyrights, and who will get the patent money? The thing is, you cannot sell it. We cannot sell it to any third party. So you can have a patent, but that's it. You cannot do much beyond it. You can synthesize it commercially in a different way, but you cannot use the living resources to get the compound. However, in the Arctic, it's a different scenario because Arctic is surrounded by countries who have got an international boundaries, ending up in the Arctic Ocean. So they have their own boundary, international boundaries over there. They are free to explore, they are free to exploit the scenario. However, because it is a very pristine environment, there are restrictions in how much you can do and whether commercially extraction of living resources is possible or not. And there are some restrictions, but actual commercialization of some of the research has been done, and it is going on in a significant basis for the last almost 5 decades. So, as I said, the Arctic, Southern Ocean, and Antarctic are extreme environments, so it's an ideal region for extremophiles and looking to bioactive molecules. And you can look for cold active enzymes, which are of industrial importance, bioremediation, molecules for bioremediation and other related technologies. Antifreeze proteins, that's a place where you can actually go for antifreeze. Antifreeze proteins, most of the organisms will have it because they need to survive under sub-zero conditions throughout the year. Lipid studies like polyunsaturated fatty acids and other compounds, which are dietary supplements, and those who are looking for pharmaceutical compounds, going into actinomycin-based research or other microbes for pharmaceuticals. There also, it's a good venue for those kind of research. So the microbial diversity in Antarctica, as I said, microbes in natural environment is not strictly bacteria. So anything that comes under the microscope is a scope for microbial exploration. So you have bacteria, we have fungi, we have algae, we have viruses, we have other protists which are microscopic, maybe few micrometers in dimension. And I mentioned here actinobacter separately because that's something that is very catchy for the people who are interested in biotechnology. And these are the different aspects I already mentioned before, polysaccharides, biosurfactants, proteins. So these kind of research is being carried out in Antarctica by various countries, but even the patents that are being put up are based on the processes, not on the organism per se. So here you can see bacterial diversity studies. Now recently we had come out with a chapter on polar microbiology and its importance for biotechnology. And in that chapter, we have actually reviewed all the literature, and can see that diverse sources of microbial habitat like seawater, fast ice, frozen lakes and ponds, rocky surfaces, microbial mats, sediment samples, even permafrost samples you can see, and symbionts of sponges and other annelids and other organisms. Diverse work has been done to understand the diversity of the microbes, and it is unlike what we consider that it's a frigid area, so you might not find a lot of microbes in that scenario, but it's not like that. It's a very diverse environment, and those who are interested in microbial diversity studies is something that is waiting to be explored, tapped. And these are the list of bioactive compounds which have been isolated from Antarctic microbes, whether it is antibacterial, anti-tumor agents, immunosuppressive agents, therapeutic enzymes. So you can see the range of compounds from range of microbes, but you can see that actinobacter form a huge chunk of it. Then you have got Pseudomonas, you've got Cardia, Penicillium, etc. So yes, people have been working on this area for a pretty long time, and we have also worked on the enzymes reported from Antarctic microbes. So you can see this alpha amylase, galactosidase, chitinases, lipases, esterases, proteases. So basically, what people normally look for in any other environment, you can also find it here. The only added advantage would be that if you are going to operate it under sub-zero, any process which is under sub-zero conditions, these enzymes are more responsive than what you find in terrestrial environment in a tropical scenario. In this Indian sector of Southern Ocean, where I normally work, now we have been working on the microbial diversity, and we are also focused on some of the interesting biotechnologically important compounds like the siderophores or ion binding organic ligands. So on the right side of the panel, you can see that these are the equatorial waters, 4 stations on this side and 8 stations in the Southern Ocean. We sampled these waters up to the depth of 200 meters below the surface water at 3 distinct depths, and we carried out the microbial diversity studies in this region. And you can see here that on the panel you can see that this is the surface samples. This is the deep chlorophyll maxima. And for those who are not aware of what is deep chlorophyll maxima, when you look at a vertical profile of chlorophyll, which is the primary source of nutrients for all the heterotrophs, the algal growth moves from the surface to slightly deeper depths depending upon the intensity of the sunlight and availability of nutrients, etc. So sometimes you'll find that the chlorophyll, although we expect that where the sunlight is maximum, you should get more chlorophyll, but it's not always like that. The chlorophyll maximum concentration will be subsurface, maybe at 30 meters, sometimes at 50 meters, sometimes as deep as 70 meters and 80 meters. So yeah, ideally the question is why they are there. It's mainly because either the light is too intense, it's too much of UV that most of them cannot survive on the surface, so they migrate to the subsurface level. Nutrient availability is more in subsurface level, so they will be there. So basically, there are numerous environmental conditions why they are always they're generally found at slightly deeper depths, and that we call it DCM or the Chlorophyll Maxima, and at 200 meters. So basically, you can see here, in general, proteobacteria is a predominant, whether it is equatorial waters, which is this 4, or the southern ocean waters in this side of the panel. So you can see that there is a shift in the concentration of the abundance of the organisms. So proteobacteria is predominant in almost all, but as you go further down south, you'll also find more contribution of cyanobacteria, and then you will have also of actinobacteria. So there is an increase. Again, it varies with the depth. So there is a difference in the depth, community differences there, and spatial differences you can see. Now, this is just to give you an idea that when we do a statistical analysis and do a Venn diagram of that, we find that almost. So this is the surface. This is the DCM, and this is 200 meters. So you find that across the different latitudinal waters, you'll find that 21% is common for all these waters. Whichever depth you go, you'll find that 21% is common. Whereas approximately 9% is unique for the surface. So again, that is controlled by the temperature, and salinity, and other environmental factors. When it comes to the frontal waters.
Dr. Bhaskar, 1 minute. Sir, you have 5 minutes more to complete it.
So let me go ahead. So we have isolated the siderophore producing bacteria from these waters, and you can see this panel. They're all isolated from the equatorial waters as well as the southern ocean waters, and the diversity we can find over here, the culturable bacteria. And again here, we also found that there are some unique organisms which are found only in the tropics and not in this polar region, and vice versa. So most of this produce more than 10 to 50% of siderophore, and the siderophore have got biotechnologically important. We are also in collaboration with Satyabama University of Chennai, and we are working on the actinobacterial diversity here, and we have just finished the data processing, and we have submitted this paper from sediments and surface water from the water samples in this station and the sediment samples in this station. So further studies are being done on the biotechnological potential of these actinobacterial isolates which have been obtained from these samples. In the Arctic also, we have been working on the diversity of the bacteria, and we find, and these are the stations from where we have collected the samples, and you can see this on the panel, right and left hand side. Again, proteobacteria is the dominant, whichever area of the globe you go in the water, proteobacteria is the predominant bacteria. And what we found is that organic matter like carbon, carbohydrate, protein, they all influence the bacterial community structure, and also the kind of bacteria that is growing in association with the algal community. So in the Arctic, we also carried out another surface sampling in this region, and it is very critical because it's an environmentally sensitive area. So what we found was that Synechococcus dominance in some areas was clearly observed as compared to total bacterial abundance, and there was a negative correlation between the bacterial and the cyanobacterial population. And we also carried out some work on the impact of temperature on algal cells and how it affects their physiology. The reason is that the same physiology effect is seen in other organisms which are feeding on this, and it will have an impact on the microbes which are going to make use of this algal biomass. So this panel shows about the bacterial siderophores, which we had isolated in the previous study. And it shows that pH and ion concentration affects the siderophore production, especially lower pH affects the siderophore production and the growth of bacteria, whereas a higher ion concentration, the 50 micromole, affects the siderophore production, like concentrations. And siderophore producing bacteria, certainly we think that one or two kind of siderophores, but we found that they produce more than one type. And Cyanobacteria was found to be the most dominant. And all the cultures, and even in the field, we found that this Cyanobacteria is predominant. And these are the types of siderophores which are isolated from these different bacteria. And these are some of the data that got from the cultures, the siderophores we found. Antifreeze proteins is the next set of biotechnologically important molecules which we are now initiating the study. And you can see this is the way the antifreeze protein acts in thermal hysteresis of the ice crystals in these organisms. And they are obtained from various sources, including fish, bacteria, and fungi. And these are some of the outputs from the.
Time limit is
Just give me 2 minutes. I'm finishing.
Oh.
These are some of the results that we have got from various other studies carried by my colleagues in the institute. And these are the diagrams with the EPS, you can see, and these are the bacteria with the EPS, and it can be used for aggregation of marine litter or microplastics and its degradation. So since microplastics is one of the important so here I'm just I've rushed through this now. So basically, those who want to involve themselves in microbiology and the biotechnological aspects, it's a multi-institutional, multidisciplinary work. So we always look for active collaborations when it comes to microbiological and biotechnological research. And presently, we are having collaborations with these many universities. Yeah, so I think that would be, yeah, thank you.
Thank you, Dr. Bhaskar. There's a time for questions, one or two questions from the audience.
Actually, I was told that the talk will be for 45 minutes, so I had prepared accordingly. But if you have told me in the beginning, I would have cut off the slides in the beginning. Anyway.
So Dr. Bhaskar has given an excellent presentation about the polar microbiology and its applications in biotechnology. For audience, if anybody want to ask some questions, they can ask.
Dr. Bhaskar, can you hear me, sir?
Yeah, I can hear you.
This is Prakash Alaja from CFTRI. I just wanted to know with reference to the diversity and culturability of a microorganism which are found on the polar.
Yeah. So what exactly would you like to know about it?
Yeah, that how the rich in the diversity they are and how difficult it is to cultivate them in a laboratory condition.
Yeah. See, one aspect is that although we consider polar regions to be something where the microbes will be adapted to the cold environment,
exactly.
But what we found is that many of them are mesophiles. They can actually grow much better under our regular laboratory conditions. So most of this bacteria which we cultured, they were growing at 20 degrees. Okay. So you will, but if you are strictly interested in psychrophytes, which are very slow growing, which they are right now dealing with with Satyabama University, they are actually looking into psychrophilic actinobacteria. They are very slow growing. It takes months for them to grow, and to get a compound from them of of noticeable level, you will have to actually keep them growing for a long time, and the biomass that you may get may not be enough later on for any kind of application. So normally, the advantage of a mesophile, if you isolate from there, is that you can grow it under low condition, and it will still grow. And it is not that its activity gets suppressed; it still grows well. At 4 degrees also it grows, and most of the activities are seen. But what happens with the psychrophile is that you really have to keep it at a very low temperature for a long time in order to get the threshold biomass from where you can actually carry out some bioactive molecule study. And that takes a lot of time and also is prone to contamination after a period of time, because unless we have a very dedicated laboratory facility for those kind of organisms, it becomes difficult to maintain them in the laboratory in our conditions. So we need to have a cold laboratory, which right now with Satyabama University we are actually developing this cold laboratory wherein they can grow the psychrophytes, especially the actinobacteria. This is psychrophytes. So yeah, there are limitations, but it doesn't stop us from looking into the microbes in a different way, in the sense that they can grow at 15 degrees or 10 degrees. There is no reason why we cannot exploit or look at the possibilities of looking at those organisms, because they grow very well at 10 degrees, even though they might be taken from a sea ice which is sub-zero. But they grow very well at 10 degrees. So 10 degrees is the temperature that we can maintain even in India, anywhere.
What about the bioprospecting? You adopt a metagenomic approach, or you use
Actually, I was going to show a slide over there. That the conventional method is okay. You try the old method was isolate and then look for the compound and then you see the activity. But now they are looking at the metagenomic method.
Okay.
That you do the whole genome analysis, look at the specific genes that you need to look for targeting, and then I target those organisms which harbor those genes and then directly go for it, rather than taking all these n number of isolates and then maintaining them and exploring each one separately. Instead of that, genomics is the new way to go, where you can do a whole genome analysis of the isolates, look at the cultures which are having the targeted genes which we are interested in, and focus on those isolates and leave the rest. That way, actually, you can speed up the whole process. Yeah.
Thank you. Thank you.
Any other questions? Good evening, sir. Jay Shankar speaking. Hello.
Hello.
Bhaskaran, sir.
Yes, sir.
Yes. Our chairman, Dr. Kunashri, is extending a vote of thanks to you. Please be online.
Yeah, thank you.
Yeah, good evening, all of you. I would like to extend my thanks and gratitude to Dr. P.V. Bhaskar, sir. Sir has given an insight about the polar microorganisms, diversity of polar microorganisms and their applications, biotechnological applications through metagenomic analysis. I think this talk would have enlightened all of you and all of us with a lot of knowledge. So I would like to extend my heartfelt thanks to Dr. Bhaskar, sir, on behalf of Department of Microbiology PG Center on behalf of Mangalore University and my own behalf. I thank you, sir. Thank you very much, sir.
Thank you. I also apologize that I could not make it physically over there, because I know that in such conferences it gives us a once in a lifetime chance to meet people whom we normally will not come across that easily. For example, listening to Professor Dayananda Agzarsar's talk was really interesting. Collaborating with Syed Dastigar of NCL. So it was really interesting to see the growth of Dr. Syed in Gulbarga University and how he has come up, and also the kind of work they have been doing. So personally, nothing beats offline conferences like how we are holding it now. Online is just to make sure that I'm not missing anything. But offline would have been much more fun for me, and I would have been really lucky to meet all of you.
Sir, shortly you will be invited for the physical presence here and for another invited talk, sir.
Sure.
Thank you very much. Thank you.
Good evening, everyone. Now we are beginning with oral presentation by PG students and research scholars. I thank Dr. A. Jaykumaran sir and Geetanjali ma'am.
Dear friends, we have a oral presentation by 12 members now. The organizing committee has decided to give a best oral presentation award and also best poster presentation award. Therefore, an evaluation panel has been formed. Professor Dayananda Agzarsar, Honorable Vice Chancellor, Gulbarga University, Professor A.J. Nair, and myself. We three members will be in the panel for the oral presentation. And the presentation will be 4 minutes plus 1 minute. Here the marks will be also consider for the timings. Even if you take extension of 4 minutes or less than 4 minutes, that also matters. I request all of you to kindly note down, and all the best.
I request oral presentation panel members, Dr. A. Jaykumaran Nair sir and Professor Dayanand Agdar sir to take over the session. First participant, Akshat US. Next participant, Damini BM, Beard.
Good afternoon, all. Honestly, I was not aware of competition. Well, I'm Dr. Akshat US, and I work at Nite University, Center for Science Education and Research, NUCSER. So the title of my topic is Dissecting Multivalent Viral Receptor Carbohydrate Interactions Using Polyvalent Multifunctional Glycan Quantum Dots. The project code name is DC-Sign MFN. So this, coming back to the host pathogen interactions, the general mode of interaction of host pathogen is through the dendritic cells, which are basically APCs, antigen presenting cells. So the host pathogen interactions at the dendritic cell happens through the proteins. They're not just physical binding, it's the structural function interaction, which is happening at the surface of the dendritic cells. So the dendritic cells has these surface proteins, which are DC sign and DC sign R. There are two proteins, dendritic cell receptors. One is DC sign and DC sign R. So these proteins are the one which recognizes those carbohydrates on the surface of the pathogens, which could be virus or could be bacteria. Now, these two proteins share approximately 80% of the regions they're conserved, but then their binding and transmission properties are totally different. Some of the DC sign will transmit HIV, HIV1, HIV2. Some of the DC sign R doesn't transmit Ebola, it transmits only the HIV virus. So there is a very big difference on the binding and transmission mode of these two proteins. But unfortunately, there are no structural studies of these proteins because they are surface proteins, they are transmembrane proteins basically. So it's not easy to isolate them, and their molecular weight is around 46-48 GADA, which is less for the cryo-TEM. So it's not being characterized. So this is how the protein looks like. So this is the carbohydrate recognition domain, CRD, where the pathogen come and binds through the carbohydrate interaction. Next slide, please. Well, as I said, DC sign and DC sign have very different transmission properties, binding and transmission. So if you see HIV glycoprotein 120, it has approximately 90 N glycans, and the interspike distance is about 23 nanometer, while the West Nile protein has just one glycan with this distance. This explains the distance between those glycans and binding to those carbohydrate recognition domains and transmission. So, but even after 2 decades, there is no information about the structure function. All the function is known, structure is not known. Next slide, please. So we explore the possibility of study the structure function relationships. Now, to mimic the host, to isolate the protein, the membrane protein, the proteins, and then use nanoparticles, glycans, which can mimic the pathogen. So we
So we developed nanoparticles, nanoparticles, and we coated those with the mannose ligands. See, the mannose ligands, dimannose, trimannose, multimannose ligands, these are the ligands that are involved in the host pathogen interaction. So we coated these nanoparticles with the mannose ligands, and we saw how it happens. Disease and disease both are two different proteins, but it's already been shown that binding and transmission properties using the quantum dots what we study. So we know that the binding of quantum dots to disease is very tight, but not with the disease signal, although both of them are displaying the same ligands. Next slide, please. So we went on to explore what's happening like that. It's proposed that the disease binds like this, right, to bind the CRBs, whereas the disease R binds the same place. So it doesn't bind strongly, which means the distance between these two ligands plays very important role on binding to these, and the distance between these two should match to this. CRBs, only then it binds slightly. So we developed gold nanoparticles, ligands with multiple ligands, and then we studied how those binding will affect the how the binding affinity changes. Next slide, please. Well, this is I'm not going to go to this. So the binding affinities where GLP use mannose ligands to dicycline and dicycline. You can see that increase in the mannose ligand surface density on the nanoparticles will give increase in the binding affinity, which means it is matching the ligand density on the surface, mimicking the host pathogen interaction. Whereas dicycline R, as I said, it's not slide, it's binding sideways. So the affinity is very, very less, which explains why some of the viruses are transmitted through dicycline and some viruses are transmitted through dicycline because of this distance. Next slide, please. This was published in JACS. And then we studied the HIV and Ebola, taking the case of HIV and Ebola virus. As you can see, when we these are the controls, and when you saturate those cells, HK293 cells, with the ligand-coated nanoparticles, the virus cannot basically come and bind, because HIV and Ebola binding sites are already being saturated with the nanoparticles. So they can't bind, so thereby the express level goes down, and we saw the same things for nanoparticles with different ligands, different ligand densities. And as you can see, the dicycline R binding affinity has come down to a drastic level compared to that of the earlier result. Next slide, please. Well, I don't think I have time for this. Next slide. Well, we wanted to make not just the distance between the tetramers, but to mimic the entire virus. So monovalent interactions are not doesn't happen in biology. It's multivalent interactions. So we made coercion satellites using the nucleic acid assemblies. It's called as nucleic acid induced nanoparticle assemblies. We made different assemblies of nanoparticles using nucleic acids so that we can match the glycan the CRD distance. Next slide, please. Well, this is the cryo-TEM image of thinking of those nanoparticles. So you can see this, there are two nanoparticles linked together. So we call it as dimers. There are many dimers over here. And once we made this dimer, the binding affinity with dicycline R changed dramatically, which is approximately 36-fold increase compared to that of QD with unlinked nanoparticles. This is because it's matching the binding distance of the CRDs. This information is very, very crucial to design synthetic glycoconjugates or vaccines where the antigen host pathogen interactions can be blocked. Well, this slide costs 2 lakhs, 2.5 lakhs. The cost of one cryo-TEM is around 1 lakh. So it's a very costly slide. Next slide, please. Well, what currently we are doing is we are at Luxor. This is what we are doing. Nanoparticle-induced assemblies to study dengue virus interaction, the host pathogen interaction. We are using glycan nanoparticles to provide structural function relationship with dengue with the dicycline R, same as we did for HIV and Ebola. And we are seeing that can these multifunctional nanoparticles mimic the host pathogen interactions. These are our collaborators. Next slide, please. Acknowledgements, Rajan and Bruce, University of Leeds, Stephen from German Private Center, Nicole, University of Leeds, Vicinity, who is working on host pathogen interactions for dengue, and these are our funders, collaborators. Thank you.
microbial synthesis over. We are keeping it now around 3 to 5 nanometers, considering long-term application, in vivo application of this, because that has a renal clearance. So we synthesis by seed mediated method.
Next, Ganana B. Shetty.
So, good evening, everyone. Myself, Damini DM. I'm an MSc student of Biotechnology second year from Karnataka University, Dharwad. So, today I'm going to present our project work related work which we have done in our labs. That is phytochemical screening, GCMS, UV-Vis, and FTIR analysis of leaf methanolic extract of Sapindus mucorrhozeae. Next slide, please. This project was done under the guidance of Dr. A.B. Vedamurthy and co-guide as Pooja R. Ma'am. So the contents of my presentation are introduction, materials and methods, results, and the conclusion. Next slide. So introduction. Nowadays the traditional medicinal system of medications are given to cure various diseases. So now we have used the plant Sapindus mucorrhozeae in our project as it has the importance of curing many diseases related to eczema, pimples, as well as psoriasis. So Sapindus mucorrhozeae is a large deciduous tree which is found in all the Western Ghats, as well as it belongs to the family Sapindaceae and is commonly known as Rita or Aretha. Next slide. So the pharmacological applications of Sapindus mucorrhozeae is it acts as an anti-cancer, anti-ulcer, anti-dandruff, anti-helminthic, and antioxidant, as well as anti-inflammatory in function. Next one. So the study site, that is from where we have collected the sample, it is collected from the Targal Huligal region of Sirsi district, Sirsi Taluka, Uttara Karnataka district and Karnataka state. Next slide. So the methodology. So what we have done is we have collected the sample, that is leaves that was surface sterilized and was subjected to shade dry, and then we have coarsely powdered the dried leaves. The coarsely powdered dried leaves was then subjected to extraction using a Soxhlet extraction method. After the extraction, the extract was subjected to phytochemical analysis and further the separation of the biocompounds which was obtained from the phytochemical analysis was done using TLC, that is thin layer chromatography. After this, to confirm the presence of the bioactive compounds as well as the secondary metabolites in our plant, we use various analytical techniques such as GCMS, UV-Vis, and FTIR. Next slide, please. Next slide. So the first step, that is collection of plant sample. As I said, I have collected the plant sample from the Sirsi Taluka of Uttara Kannada district. Then we have surface sterilized the leaves using 0.01% of mercury chloride and ethanol. Later on, we shade dried it and then we have formed a coarse powder of that. Next slide, please. After that, the preparation of extract. The extracts were obtained by Soxhlet extraction method using a constant hot extraction technique. Then we have done a phytochemical analysis using various chemicals to know the presence of the secondary metabolites in our plant extract. So some of the tests were alkaloids, we have done Mayer's test, and saponins is the main, that is foam test. Next slide, please. After this, we have done a thin layer chromatography to separate the compound which we have obtained from the phytochemical analysis. The mobile phase which we have used is a chloroform and methanol, that is 90:10 ratio, and the stationary phase was silica gel. Next slide. Then Coming to the analytical technique, nowadays there is a dramatical increase in the technology. So we have used some of the powerful tools such as UV-Vis, FTIR and GCMS to confirm the presence of the compounds which we have found in phytochemical analysis. So the first comes the UV-Vis and FTIR spectroscopy. UV-visible is used to know the presence of the secondary metabolites at 200 to 800 nanometer estimation. Then FTIR is done by 1000 to 4000 nanometer absorbance, which shows the functional group as well as the bonds present in our compounds. Next slide. Okay, in GCMS is the analytical technique. As you all know, it is a gas chromatography mass spectroscopy. In this we use the helium gas for constant flow rate, and the software adopted was turbo mass, and we have interpreted the GCMS using results using NIST, that is National Institute of Standard and Technologies. Next slide. So the results, what were the results we obtained was the plant extract was obtained from the area of Circe. Then the Sapindus mucorosae had a functional metabolite, that is alkaloids, flavonoids, saponins, etc. Then by TLC we found the RF value as 0.92, and there were several phytochemical analyses were also done. Next slide. So in next slide, that is we found that in gas chromatography, the crude extract reveals the more amount of bioactive compounds, and we got to know about the melting point, the structure, and the ligands or the compounds which are present in our plant extract. Next slide. This is all about the GCMS only. We have found 45 compounds in our plant as a bioactive compounds. Next one. So in UV visible analysis, the occurrence of peak reveals the presence of secondary metabolites in the plant extract. So as I told, it reveals about the presence of secondary metabolites in our plant extract, which was confirmed using UV visible. Next one. So in FTIR, the phenols, alkanes, alkenes, as well as some of the functional groups was estimated. So the conclusion is that, as I said, in GCMS they found about 45 compounds, and in further studies there should be needed to undertake the isolate and screening of bioactive compounds from methanol extract of aerial parts and find out its biological activity. These are the references which we have used in our studies for the extraction of the bioactive compounds. Next one. So thank you everyone.
Ramila. Next, Ishrat Jahan. Good evening, everyone. I am Pramila Iparthi, working under the guidance of Dr. Prakash Hadami from CSAR CFTRI. Next slide, please. We all know that fermented foods are known for their health benefits, which enhances the shelf life of the food product and increases the flavor. Nowadays, we all know that it has become an integral part of our daily life, and not only in the fermentation process, but the microorganisms which are involved in fermentation provide equally for the benefits of our health.
So mainly the northeast regions like Sikkim and Manipur people prepare indigenous fermented foods like kinema, hawaiian, and they are nothing but the fermented soya bean, which are mostly dominant with bacillus species. So they produce some small heat stable peptides which are known as bacteriocins. They are having biopreservative agent as a potential application. Next slide, please. So we all know that the bacteriocins are the ribosomally synthesized peptides which are post-translationally modified. And coming to the term antibiotics, it contains lanthinin containing antibiotics. Lanthanins are the unusual amino acids produced mostly by gram-positive bacteria. So subtilin and subtilin-like antibiotics comes under the class 1 of antibiotics, which are mostly divided into 4 classes of antibiotics. These subtilins are having a sequence of 32 amino acids, and sublichinin is a new subtilin-like antibiotic which is produced from Bacillus licheniformis. So the other strains producing subtilin-like antibiotics are Bacillus subtilis and Bacillus spicezani. So sublichinin is an amino acid. It contains a sequence of around 56 amino acids, which was newly identified by in CSAR, CFTRA, and subtilin A is a bacteriocin which is having 43 amino acids, mostly produced by Bacillus subtilis strain. Next slide, please. Here is the structural presentation of all the antibiotics which differ from each other with maximum 3 to 4 amino acids. Next slide, please. So here is the workflow of our plant where we have isolated bacillus cultures, checked for their antimicrobial activity. Further, they have proceeded to check the mode of action by using specific reporter strains. Reporter strains are nothing but the genetically modified organisms which help in the rapid screening and identification of subtilin-like antibiotics produced. So here is a representative plate presenting both the positive and the negative controls of the test strains. So here blue color around the culture strain indicates it as a positive strain, where absence of the blue color indicates as a negative control. Next slide, please. Further, we have performed, we have screened around 92 fermented food samples from various northeast regions, out of which we could find where maximum beta-galactosidase assay was shown by the strain SA3.7, where it quantitatively measures the amount of beta-galactosidase present. Indirectly, it tells us whether subtilin is induced or not. Next slide, please. And we have submitted the culture for genome sequencing, and we have identified the strain belongs to Bacillus subtilis. Next slide, please. We have carried our gene-specific PCR where we have found that the amplicon size of this bias gene, which is responsible for subtilin-like antibiotic, is present. Next slide, please. Further, we have carried out the fermentation or the purification of our peptide. Here is the flowchart representing, and here is the page which consists of the molecular weight of our peptide belongs to nearly 3.5 kDa. Next slide, please. And further, we have done mass spectrometric analysis. Next slide, please. And we have found that the fraction contains around the subtilin, sublichinin, and which are also succinylated, which are found to be a kDa of around 3 to 3.5 kDa. And interestingly, a unique cyclic variant of subtilocin is also present. So here we can prove that our strain can be used as a fermentative and also in the preservation process of the food product. Next slide, please. Thank you.
Next, Krishna Komatavar, be ready.
Ishrat Jahan.
Good afternoon, everyone. My name is Ishrat Jahan. I'm from CFTRI, Mysore. I'm working under Dr. Prakashan Halami. It is my pleasure to present my piece of work in today's National Conference on Integrated Microbial Technology for Sustainable Future. To start with my topic, the production of antimicrobial peptide is strongly influenced by the media composition, such as it includes carbon source, nitrogen source, and minerals and salts present in that. And the media components and their optimum level is very crucial for the optimum production of the high influence production of the antimicrobial peptides. For the production of metabolites, media optimization is considered as one of the crucial steps, and it will, for the product to be economically feasible, optimized media is necessary. And Bacillus legniformis MCC 2514 is my strain where I'm been working with. This is a beautiful organism which produces an antimicrobial peptide which has inhibition activity against several food pathogens. So there are several advantages of medium optimization, such as enhanced metabolite production, cost effectiveness, organism specific developing of the media, and natural production of the post-translationally modified products, and of course time saving. What is genetic algorithm? Genetic algorithm is a method which I'm employing for my work for the media optimization. This is a method for the optimization based on the evolutionary algorithm that is like natural selection. It is same as how the crossing over, or in the DNA how the crossing over happens. It applies same, and the best species finally what we get is the best out of the best. So it has several advantages like multimodal target function, complex data sets we can handle, and accuracy prediction over RSM and any other methods of optimization, random initial cost function. So the methodology includes medium component selection. So starting at the first, the step of the any optimization process will be media component selection. So I have selected 8 media components, which I will be explaining further. Preparation of the inoculum for the fermentation process, and then genetic algorithm using a software I have done, MATLAB software. Then finally optimized media concentration we will achieve. So genetic algorithm, exactly how we will carry out. First, initialization of the population, or the creation of the population we have to do. Then the random population we will create. After getting the response from that population, fitness function will be performed using the software. That is out of the media composition, 10 media composition we had taken. Out of that, best will be selected and further proceeded. Selection is based on Rowland method, and reproduction is carried out, and the convergence will happen. Here are the 8 media components which I have taken. So these are the ranges of the media components per liter, mg per liter, and the black dot indicates the reference media where the concentration lies. And this is the graph which says the frequency of the particular range of the component of the media, which is occurring again and again in that kind of in that particular of concentration. Like lactose is having lactose in all of the 5 generation, 5 to 10 in the 5 to 10 grams per liter of the range. It is occurring frequently. So our optimized media concentration will be having in that. And then same in the NH4Cl and NaNO3. These are the 3 major components which is having high influence on the media. And here I can see the response I have recorded of all the 5 generations, where AU per ml and OD is AU per ml and OD I have taken, and this particular dot is the where I have got the highest production of antimicrobial peptide, and OD is also highest in this. And the results here also suggest the same thing. And here until the 5th generation, until the constant terms when we get, till that I have performed the experiment. And this is the final media, optimal media concentration, where I had said you that gram per liter, this lactose, NaNO3 and NH4Cl is the major things, and we have got in the same range which we have predicted. And this is the antimicrobial peptide antimicrobial assay, where against Purpurea or Rhizophila we have carried out, and ONPG assay, where it will show the quantitatively how much antimicrobial peptide has been synthesized in the media. So we have got in this, where OSM is the optimized synthetic media, we have got the highest production. This also includes in vitro same results it will depict with this. And my conclusion is genetic algorithm is the accurate method to optimize the media, and optimized synthetic media we have got for the Bacillus ligniformis especially, and antimicrobial peptide production in OSM has yielded 3 times higher activity compared to the unoptimized media, and antimicrobial peptide produced from Bacillus ligniformis is acting on RNA biosynthesis inhibition. Thank you so much. Yes, sir. It has lowest, we can randomly generate the population. It doesn't need the specific these terms only like that, it doesn't need. And it is a time saving method, sir. Compared to, even I have performed the RSM methodology, so in that it is not. Thank you.
Gamana B. Shetty. Online presentation. Start your presentation.
Hello everyone, I am Gamana B. Shetty, postgraduate student, Department of Microbiology, School of Life Sciences, Justice Academy of Higher Education and Research, Mysore. My title entitled with Clinical Assessment of Oral Probiotics Profile in Neonates and Pediatric Cases under the guidance of Dr. Sumana K., Department of Microbiology, School of Life Sciences, Justice Ahir, Mysore. Coming to the contents of my topic, what are probiotics? Probiotics in pediatrics, types of bacteria present in the probiotics, characteristics of oral probiotics, benefits of oral probiotics, side effects of oral probiotics, mode of action, can probiotics help in medical conditions, future aspects of oral probiotics, and its conclusion. Coming to what are probiotics? Probiotics can be defined as living organisms or as food ingredients containing living microbes that beneficially influence the health of the host when used in adequate number. The term probiotic comes from Latin or Greek pro, before forward, and bios or life. Thus, probiotics are life promoting. In this case, we use the term probiotics to refer to the beneficial bacteria. Concept of probiotics by Elie Machnikov in book Prolongation of Life in 1907 proposed a diet containing milk fermented by lactobacilli, which also produced large number of the lactic acid that could increase the lifespan of the humans. Coming to the probiotics in pediatrics, according to Nestle Research Center, bacteria found from mother gut are transferred via breast to the infant gut. Translocated bacteria influence colonization of infant bowel, contribute to education of infant gut. For multiple and variable reasons, for some infants cannot be breastfed. Research has proven that formula with probiotic provide the same flora as breastfeed, reducing allergies, infections, and improve defenses. Natural birth is not sterile, and neonates do not benefit from this inoculum. Diet in early life is a major factor for this ecosystem. Since breast milk contains bifidobacteria, it also contains oligosaccharides that promote the growth of bifidobacteria. Coming to the types of bacteria present in probiotics. The first probiotic species introduced into research were Lactobacillus acidophilus in 1983 and Bifidobacterium bifidum in 1991. The most common types of bacteria present in oral probiotics are Streptococcus, Lactobacillus, Bifidobacterium, Propionibacterium, Lactococcus, Enterococcus, Pediococcus, and Acidobacterium. Lactobacillus is normally found in the oral part of the body. Lactobacillus acidophilus and Lactobacillus reuteri are one of the ones that are common in Lactobacillus genera. Lactobacillus reuteri is commonly found in the intestine and oral part of the body. Yeast can also be present as probiotic. Coming to the characteristics of oral probiotics, surviving the passage through the digestive system, able to attach to the intestinal epithelium and colonize them, adherence to human intestinal cells, should be non-pathogenic and non-toxic, should utilize the nutrients and substrate in the normal diet, production of antimicrobial substances, antagonism against enteric pathogens susceptible to antibiotics. Coming to the side effects of oral probiotics. When first using probiotics, some people experience gas, bloating, or diarrhea. Changes in the gut microbiota can result in the bacteria producing more gas than usual, which can lead to bloating. In rare cases, probiotics may cause skin rashes or itchiness. Amines in probiotics may trigger headache. Some people may experience severe headache after eating this kind of foods and should instead opt for probiotic supplements. Children and adults with severe illness or compromised immune system should avoid using probiotics. Rarely, probiotics bacteria can contain antibiotic-resistant genes. They can pass this gene to the other strains, which including the harmful strains that causes infection. Coming to the mode of action. Competition for dietary ingredients has growth substrate, bioconversion, for example, sugar into fermentation products with inhibitory properties, production of growth substrate, for example, vitamins for other bacteria, direct antagonism by bacteriocins, competitive exclusion for binding site, improved barrier function, reduction of inflammation, thus altering intestinal properties for colonization and persistence within, and stimulation of innate immune response. Can probiotics help in the medical condition in children? Probiotics are widely known for their ability to prevent diarrhea or reduce its severity. Human studies find that probiotic supplements can improve some mental health disorders. Probiotics may help keep the heart healthy by lowering bad cholesterol and blood pressure. Certain probiotic strains may reduce the severity of eczema in children and infants. Early research suggests they may help the symptom of irritable bowel syndrome. Probiotics may help give the immune system a boost and inhibit the growth of harmful gut bacteria. Coming to the future aspect of oral probiotics and its conclusion. Probiotics have already been established in the treatment and prevention of the various gastrointestinal systems. Although many clinical studies have demonstrated positive outcome in preventing caries and periodontal diseases, the data is still scarce in recommending probiotics for the oral health in children. Moreover, since initial colonization of oral cavity of the newborn is very important for developing immunity and prevention of future diseases, health measures should be directed towards its preventive use in infants and children. Thank you.
Gamana, can you?
Yes, sir.
I have a small question. To give a significant message out of your presentation. Can you tell?
Sir, what, sir? I'm not able to hear you, sir.
Well, hello.
I'm not able to hear you, sir.
Now, am I audible?
Yes, sir.
Okay, now tell me in one sentence what is the message you want to give out of your presentation? What is a significant contribution out of your this presentation? So probably we are unable to connect you.
Hello. So good afternoon, everyone. Myself Krishna, and I am working as a junior research fellow under the guidance of Dr. Prakash Shalami in CRI Mysuru. So I am going to present my work on genomic analysis from the probiotic bacteria Lactobacillus fermentum, strain number MCC0552, as a potential probiotic strain. So when we talk about probiotics, it should fulfill about 2-3 aspects, like it should be consumed live, it should be in sufficient number, and it should confer health benefits to the consumer host. And it should properly colonize in the gut of the host also. Next slide, please. So starting from the basic growth parameters, growth characteristics, how it behaves, what is the temperature, like we grow it on MRS media and it is a facultative aerobic organism, gram positive, these are the basic. Then this is how it behaves on the MRS media, and that is the same image. And these are the some of the probiotic attributes of my culture, like it has acid and bile tolerance, and it has strong adhesion property, and it is known to confer health benefits by immunomodulation, like lowering of pro-inflammatory cytokines and stimulation of anti-inflammatory cytokines. So, coming to the main point, like we have done the genome analysis of L. fermentum, and it showed that the bacteria has a genome size of 22,67,305 base pairs, along with 15 ribosomal RNA, 59 tRNA, and GC content of 51.4%. So this is a circular and this is a linear map. And along with the genome sequencing, we have done the phylogenetic analysis also, and we came to know that it is also being very close to other probiotic reference strains like L. fermentum, and it has been transferred horizontally from the reference strain. So as I said, this strain is also compared with other genomes, other reference strains like IFO, F6, MTCC, and ATCC. So these are the reference strains, and in comparison, it shows that our strain has large genome size and the number of ribosomal RNA is also more, and the number of transfer tRNA is also more, and our culture has a unique property that it has 12 prophage regions. So further, we have analyzed for the functional attributes like identification of that. We came to know that our strain is able to produce vitamins like B1 and riboflavin, which can conclude that our strain has an capability of an industrial. It can be used as an industrial purposes also. So these are just the pathway, like thymine metabolism, riboflavin metabolism. On this basis, we can see that it has an industrial importance also as a not only as probiotic, but on the commercial scale also we can use this culture for the production of these vitamins. And other main attribute of a probiotic that it should adhere to the intestinal lining, to the gut strongly, that it should not flow easily, it should not go off immediately. To show it, no matter how many plenty of number of your culture has a probiotic good benefit, but to show that it should remain in the gut and confer that health benefits to the host. So for that, most important, it should adhere to the intestinal lining. So for that purpose, when compared with the other reference strains, our culture has the size of that mucin binding protein, has the largest size of mucin binding protein. Mucin binding protein helps the probiotic bacteria to adhere to the intestinal lining. So as you can see at the bottom, this is the mucin binding protein of our probiotic culture. As compared to other 4 strains, our culture has the largest mucin binding protein. So this can ensure high confirmed adherence to the intestinal lining. And about the genomic guidelines, like our culture has been compared with the other 2 strains, and it was found that it is identified the genomic guidelines with like type 2, CRISPR, Cas2. It shows that it has been horizontally transferred, like from the same parent, our culture has been transferred. It is linked to other probiotic strains also. One more probiotic attribute that it has a prophage regions. Our culture has total 12 prophage regions. Prophage region is nothing but a portion of genome wherein phage integrates its genome and it remains harmless to the host. Coming to end, these are the 12 prophage regions in our probiotic bacterial strain. And we have safe deposited this culture in NCMR Pune, under the strain MCC 0552. So, thank you.
What's the source of your probiotic strain? Dairy source. Dairy source means it's milk or some other. It's milk or something else. Which dairy source? From cow. From cow. Okay. Okay.
I request everyone to maintain silence. Next participant, Mrs. Neelam Mishra.
A very good morning to all of you. Myself Neelam from Gulbarga University. I am going to present a small part of my PhD research on rhamnolipid-assisted phytoremediation of lead-contaminated soil for sustainable future under guidance of Dr. Dayanand Aksar sir. So rhamnolipids, as we know, we are talking about sustainable future. These are the green molecules of the era. These are preferred over chemical surfactants because they are less toxic, they are biodegradable, and they have multitude of applications, as we could see here, biological as well as environmental. So first, there is a search for biosurfactant producing microorganisms from the oil contaminated sites, which is done by different screening methods. Since the screening methods are having, there are more than 11 methods and all are having some pros and cons. So here, one novel method was developed using Nerium leaf and images software, and the same work was published in Indian Journal of Experimental Biology 2021. The organism was also identified as Pseudomonas aeruginosa, DNM50, by 16S molecular sequencing. The organism was further used for production of rhamnolipids, which is a glycolipid-based biosurfactant. And the method used was, as we can see here, as we could see here, there is a foam formation, which indicates the production of biosurfactant in the form of foam. So the method used for purification as well as characterization was solvent extraction, followed by different chromatographic methods. So TLC, followed by HPLC for partial purification, then FTIR to know the functional groups present in these rhamnolipids, and then NMR, followed by the advanced method of Maldi-Top. So by Maldi-Top analysis, we can analyze that there are 12 different congeners of rhamnolipids formed here. So rhamnolipids are composed of rhamno sugar and fatty acyl chains. So depending upon its length of fatty acyl chains, we can see it's not a single molecule which is present in this compound. There are 12 different compounds which is present, due to which we could find there is variation in the properties of rhamnolipids if it is produced even from the different strains of Pseudomonas itself. So next we focus on the environmental application of rhamnolipids, which is lead contamination in agriculture soil we concern. So as we know, due to excessive use of pesticides, herbicides, we have lots of contamination in our soil, which enter in the food chain, and ultimately it affects human beings also, causing more cases of the anemia. So here, the more focus is done on the lead contamination. So if you could see here, there are many samples collected agriculture soil from different parts of Karnataka. So we could find by WHO, the recommended level is 0.1 mg per liter. But here you could see majority of the agriculture soils have shown more than the level of the, recommended levels of the lead. Thinking this, particularly phytoremediation was to enhance its what we call efficiency. So as we could see here, there were randomized block design used for the phytoremediation, and if we see there are 6 treatments given over here. We focus on D, E, and F. So in one we have used just the biosurfactant at very low concentration of rhamnolipid, which is just 50 mg per kg. And then there is marigold seeds we have used for phytoremediation, as these are known to be more efficient in phytoremediation of the lead. And another one we have used both by marigold seeds as well as the biosurfactants. So this was done in a greenhouse conditions. And if we could see here, only a small part of the results I am showing here. So if we could see as compared to the control, in the phytoremediation, the quantity of the lead has decreased from 1 mg per liter to 0.8. If it is assisted by rhamnolipids, the quantity has decreased around 0.6 to 7. And if it is only cell-free broth we have used, it is little more than the 1, which also indicates if we don't purify much our rhamnolipids, then also we can apply it as an environmental application. So we have seen a 30% reduction in the concentration of lead when we apply. So this indicates that from this study that the rhamnolipids can be a future potential molecule for the phytoremediation of the lead contaminated soil, which is also required for the sustainable future for good health of the human well-being.
How you are extracting and purifying the lipids? How you are extracting and purifying the lipids? Yeah, yeah.
It is by solvent extraction method, acid precipitation, followed by partial HPLC we have done. And then we have characterized by all different methods. Sir, I can
Okay.
There are many applications that have done on. It's a very good anti-cancer agent. In completing the things fast, I did not mention here. Actually, it is in my publication only. One of the paper is published in Frontiers in Bioengineering and Biotechnology. Impact factor is 5.89. That is the first report on rhamnolipids, where it is inhibiting something we call P38 kinases. So we say in the cancer therapy, P38 inhibition is very important. So this has shown very small concentration. It was 0.05 microgram per ml was the IC50 value. And even P38 inhibition, same compound. Our compound has given good results. Even oil washing efficacy of used lubricating oil. It has shown more than the chemical surfactant SDS. SDS has shown somewhere around 78%. It has shown 89%, more than this. So very good, very efficient molecule is there. Like that work I did not show here. I have shown only about phytoremediation. Okay, thank you, sir.
Next participant, Rana Kamath.
Good evening, everyone. Actually, I am from foreign student, so I will be very, very slowly compared with other students because in my country, we use to Arabic language, like general language. So I will talk silpa. Thank you. I will present today my subject will be etiological agent of infection, urinary tract infection. Urinary tract infection is any infection in any part of the urinary tract, the kidney, bladder, or urethra. Any infection in this part, it will be give symptoms to disease. This disease. So, urinary tract infection is one of the most common infections disease and has high financial burden as a society. The occurrence of urinary tract infection acquired by population higher and young women. Almost half of all women will have at least episode during their lifetime, and about 1 to 3 women will have at least urinary tract infection episode. So at age of 24 years, urinary tract infection increase with age for both sexes. But in the women, it's more risk compared with the men because the urethra is more shorter than males. That's why the infection will be risk more. It's estimated that 10% of men and 20% of women over 65 years of age have bacterial symptoms. So, as shown, urinary tract infection, that's the area region, causes, that's the part, it will explain the infection tract become irritated. That means it's inflammation. The objective, the aim of this review is to provide a summary and theoretical evaluation of the published evidence about the etiological agents of urinary tract infection, with focusing on the most common causes, gram-negative and gram-positive bacteria that cause urinary tract infection. We can classification this into two types by anatomic site of involvement. A, lower tract infection, that's bladder, urethra, and prostate gland, and epididymis. B, upper tract, B part layer, upper tract infection involves parachemia of the kidney. Second, we can classify the second uncomplicated and complicated. Uncomplicated urinary tract are not associated with this structure or neurologic abnormalities that may infected with the normal flow of urine or the voiding mechanism. For example, normal healthy females to 15 to 45 years. B, complicated are the result of the predisposing lesion of the urinary tract, such as congenital of the disseration of the urinary tract infection. So in the indwelling, prostatic hypertrophy, absorption, or neurologic that infection with the normal flow of the urine and urinary defense. Etiological. The bacteria that most often cause cystis and pilomenifer are the following. Interact usually gram-negative aerobic bacteria, most often Escherichia coli, 75 to 95 of cases. Klebsiella, Proteus, Marbullus, and Pseudomonas, and gram-positive bacteria, less often Staphylococcus saprophytes is isolated in 5 to 10% of bacteria in urinary tract infection. Enterococcus faecalis group Streptococci. Streptococcus group B, Streptococci. In hospitalized patients, Escherichia coli account for about 50% of cases. The gram-negative species, Klebsiella, Proteus, Marbullus, and Pseudomonas account for about 40%, and the gram-positive bacteria, Cocci, Bacillus, species Saprophytes, and Staphylococcus areas account for the remainder. Risk factors. In female, short, as I mentioned, short urethra, incomplete bladder, emptying with age, males, prostatitis, hypertrophy, bacterial prostatitis, aging, diabetes mellitus, urine retention, impaired immune system. Common symptoms. Many symptoms is there in urinary tract infection, but the important thing, abdominal pain or vomiting, nausea, and burning sensation during urine, and abnormal strong smells urine. Many symptoms is there, but this is the important things I mentioned here. Treatment. Treatment of urinary tract infection consists of antimicrobial therapy, treatment, single dose therapy, 3 days, 7 days, and 4 to 2 weeks. Single dose therapy, short course of antimicrobials such as trimethoprim, sulfametazole, and amoxicillin 3 milligram, ciprofloxacin 500 milligram, and norolofloxacin 400 milligram. 3 days therapy efficacy same as 7 days therapy with less adverse effects. Drugs used include quinolones, TMP, and SMZ, beta-lactam, antibiotics. 7 days therapy used less for in complicated urinary tract infection. So useful in recurrent cases, pregnancy, urinary tract infection with other risk factors, and for 14 days, therapy for complicated urinary tract infection, high risk of mortality and morbidity. Conclusion, urinary tract infection are the second most common bacteria infection. The most common bacteria found in case urinary tract infection is Escherichia coli. 80 to 90% women are the most infected subjected in the publication compared with other ages, different ages. Development of resistance to antibiotic by the bacteria results in problems during the treatment and led to replace occurrence. Thank you.
One basic question. Why gram-negative aerobic bacteria oftenly causes urinary tract infection when compared to gram-positive aerobic bacteria? Why gram?
Yes.
Yeah.
Discover is make more damage forward with positive. From DNA, many studies find DNA damage in the membrane.
Participant Sushant Chigan. Neha Desai. Neha, you can present your presentation.
Am I audible, sir? Hello?
Yes, yes.
PPT is visible, sir?
Yes.
Okay, thank you. I'm Sneha Desai, a research scholar from PG Department of Studies in Botany, Karnataka University, Dharwad. I'm working under the guidance of research supervisor Professor T.C. Taranath, Professor Department of Studies in Botany, Karnataka University, Dharwad. Today I'm here presenting the part of my research work in front of you, which is titled as Evaluation of In Vitro Hello? 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 Am I audible, sir?
Yes,
Yeah.
you are audible.
You can proceed.
Yes, you are audible.
Okay, these are the contents which I'm going to present in this presentation. Introduction part. Tuberculosis is a very contagious disease that is one of the top 10 cause of mortality worldwide. And the prime reason for the death caused by the single infectious pathogen. Along with this, lung cancer is also the deadliest disease which causes the lungs. And many treatments have been used to fight the cancer, including chemotherapy, immunotherapy, and radiotherapy. However, these techniques are incapable of limiting the adverse effect. So, in this regard, nanotechnology may emerge as one of the most promising options for developing the more effective medication delivery methods for TB and cancer patients. The word nanotechnology was first defined by the Norio Taniguchi, and therefore there are many methods which are used to synthesis of the nanoparticles, but the green method approach of synthesis of nanoparticle has been explored for over a decade as an environment friendly synthesis methods. Plants contain an array of phytochemicals that serve as a converted coating agent as well as a stabilizer for transporting the metal nanoparticles from metallic ions, which are resulting in the generation of preferable nanoparticles. And also I have used my study the Grevia tulifolia plant to synthesize the silver nanoparticles, which the compounds which are present in the Grevia tulifolia leaf extracts are alkaloids, saponins, tannins, triterpenoids, carbohydrates, phenols, sterols, flavonoids, and quinones. Some of the main active compounds are fridolin, lupin, and betylene. Compounds like gulonic acid, violactone, and d-erythro-2-hexanoic acid, violactone, are found in preliminary phytochemical study. The objectives of my study are biosynthesis of silver nanoparticles using Grevia tulifolia leaf extract, characterization of synthesized silver nanoparticles, and evaluation of biological activities of synthesized silver nanoparticles. This is all about the material methods which I have used. I have used the leaf of the Grevia tulifolia plants for synthesis of the nanoparticle. This is the oral flowchart of the synthesis method. And next I have used the characterization techniques. These are the characterization techniques, includes the UV visible spectroscopy, AFM, FTIR, XRD, zeta potential, and HRTM analysis. Further, for biological activities like anti-tuberculosis, antioxidant, and anti-cancer activity, this is the flowchart that I have used to evaluate these are the activities. Anti-tuberculosis activity I have done on the Mycobacterium tuberculosis by MABA assay, where risazorin dye, which is blue in color, which get converted into the pink color risaruffin dye if the bacterial cells are viable. In antioxidant activity, I have followed the DPPH assay, whereas the anti-cancer activity I have done with on the lung cancer cell lines, which is A54 line cell line by MTT assay. Here the yellow MTT will get converted into the purple formazole by the viable cancer cell. If the cancer cells are dead, then this yellow MTT will remain the yellow in color. So coming to the results and discussion part, here UV visible spectroscopy, we can see the here absorption spectra of the AGNPs, which shown the 409 nanometer, which is observed here, confirms the formation of the silver nanoparticles. Broad peaks at lower wavelengths usually imply the rise in the particle size, while the narrow peak at shorter wavelengths indicates the development of the smaller nanoparticles. Coming to the next is AFM analysis, that is atomic force microscopy. Here the image depicts are the two-dimensional representation of the monodispersed spherical silver nanoparticles with an average size which is ranging from 21 to 65 nanometer. Coming to the next is zeta potential analysis. Here we have found that minus 35.8 MV zeta potential, which are the stable nanoparticles in the colloidal solutions. And also in the FTR analysis, we have seen we can see here the FTR analysis of AGNPs as well as the leaf extract. These are the absorption peak and their respective functional groups. Here, phenols, leaflet proteins, and ketones, alcohols, and phosphatanes are involved in the synthesis process. We can see here clearly. I've also capped the silver nanoparticles which are synthesized. Then next coming to the XRD graph, here we can see the 5 2 theta degree values, that is 38, 44, 64, 77 and 84, which are referring to the 5 different planes of, that is 111, 200, 220, 311 and 222, that is 222 respectively. Also this data has matched with the standard JCPDS card number 0000040783, which confirms the crystalline nature of the nanoparticles synthesized. Coming to the next characterization technique is stem analysis, where we can see the spherical shaped nanoparticles here. Also, we can see the lattice fringes and size, which is size ranging from the 11 to 34 nanometer nanoparticles. Also, in the shade pattern, we can see the inner ring. Here it is present. That is one inclined to the 111 plane, a characteristics diffraction ring of the AGNPs. Coming to the biological activities, the first one is anti-tuberculosis activity. These are the standard drugs which I have compared with my two samples, that is AGNPs, which are synthesized, as well as the plant extract. Here we can see I have found out the MICs of those all samples. Here we can find the AGNPs have showed the minimal inhibitory concentration as 6.25 mg per ml, whereas in the plant extract it is higher, that is 50 mg per ml. These are the standard drugs which I have used for the comparison: isoniazid, ethambutol, pyrazinamide, rifampicin, as well as the streptomycin. And also we can see here the pink color where the bacteria are growing, whereas in the blue color here the bacteria are dead because they are not converting the resorzurin dry into the resorphin dry.
Neha,
Hello?
time up. Make it fast, please.
Yes, yeah, ma'am. Only 2 minutes. Coming to the antioxidant activity, here we can see the antioxidant activity of the 3 samples. I have compared all my 2 samples, plant extract and as well as AGNPs, with the standard ascorbic acid. Coming to the anti-cancer activity, here we will see the microscopic images of in vitro anti-cancer activity on the lung cancer cell lines. I have compared my AGNPs as well as the plant extract with the cisplatin standard drug. We have found out the IC50 value of AGNPs is 23.45 mg per ml, whereas extract is 56.31 mg. So here nanoparticles have shown the greater activity than the plant extract. Coming to the conclusion is that this is the first study to show an capability of the leaf extract to Grevia telefolia as a reducing and capping agent. Also, the synthesized nanoparticles have shown the anti-tuberculosis, antioxidant, and anti-cancer activity. And we conclude that Grevia telefolia leaf extracts can be used as alternative to medications in the pharmaceutical applications. I also thank the chairman as well as the safe USIC in the Karnataka University, Dharwad, for providing the laboratory facilities and stick coaching for HRTM analysis. Also, I thank the Maratha Mandal Dental College, Belagavi, for providing the laboratory facility for doing the anti-tuberculosis activity. Also, I thank the Mangalore University for conducting this webinar so that I can present my part of the research work. Thank you very much. These are the references. Thank you.
Neha, am I audible to you?
Yes, sir.
So I have a one question.
Yes, sir.
Basic silver nanoparticles make them as anti-tuberculosis agent.
Yeah, sir.
Could you get my question?
No, sir. Please pardon, sir.
What are the characteristics of silver nanoparticles
Yes, sir.
make them as agent?
Actually, it is inhibiting the bacterial growth, sir, at 6.25 mg per ml. That is the characteristics. Also, the further studies are also required to see the whether exactly where the nanoparticles are attacking the in the tuberculosis. So that study is going on, sir. It is only the part of my study, sir.
Please listen. Why do they do so?
They are very smaller in size, sir. Silver nanoparticles are very smaller in size. They can
If they become smaller in size, what properties it gains? Anything which is smaller in size can do the anti-tuberculosis agent?
No, sir. They easily disrupt the cell wall and membrane of the bacteria, as well as disruption of DNA. They can do. Sir.
What that special property makes them to do so? It's a very basic question. Anyway.
Thank you, Sneha.
Participant Tejasvi B. Shetty.
Good evening, everybody. Myself Tejasvi Shetty. I'm research scholar. I'm presenting the topic on the documentation of grasses on Kodagu. So I'm doing my research work under the guidance of Professor M. Jaishankar sir. Yeah, next. So coming to the very importance of grasses, these grasses that is poesy, I can say that we can just bow down, it will be not exaggerated if we bow down, for the poesy family itself, because they are the major part of the staple food. So, next. So the very topic that is with the documentation of grasses, actually, we are very much utilizing the identification characters, that is the parts of these grasses are main, basic knowledge of these is very important for the identification of grasses actually. So next. So my objectives is the collection, identification and documentation of grasses. So very much, this is just to show that here, where the poesy stands actually, where it is its status actually. So in India, all over the world, it is 12,000 species of poesy has been identified under 780 genre, whereas in India, it is just 1,506 species under 266 genre, and in Kodagu actually very less work has been done actually when compared to the dicotyledons, monocotyledons. The basic work has been very much lesser actually. So where only 135 species has been identified by Yogananda Simha sir and Keshav Murthy sir in 1990, and the another work it is from B.C. Banerjee that is 1985 identified the new 4 species actually to the Kodagu district actually. Next. So this is the area of study that is Kodagu district itself. So various different places of Kodagu have been selected for the collection of these grass species. Next. So here in the very simplest methodology was been carried the collection of grasses from the site and basic noting of these every collection points actually, the field notes with notes that is very much with the site identification, locating with the GPS help and all and yeah. Herbarium was prepared actually of each specimen, whatever been collected actually. Fresh samples are dried with the blotting paper and newspapers will also be used actually. The herbarium preparation, you know that. Then all about 68 herbarium specimens were identified actually. So yeah. So all the 68 herbarium species have been documented in DOS, Mysore University, Manasa Gangotri, which been assigned with the OCHA numbers. So it is identified, it's a confirmation for the identification and it is the authentication for its identification, of course. I should thank Dr. Gopal Krishna Bhatt sir, the late the retired professor of Purna Pragna College, who helped me in the identification of these grasses actually. So these are the herbarium specimens, a few samples that's all. These are the identified 68 grass species actually from Kodagu district itself in 2 years span actually. So the voucher number is also put in there in the last column. So these are the field images, photographic images of these collected samples of mine. So this is just to show that where it stands actually, the very tribe of the family, that is the total number, the relative abundance we see with respect to its genera and species in terms with respect to the tribe of this poaceae family itself. So that is all. Thank you. I should thank our guide, Professor M. Jaishankar sir, also for guiding and supporting throughout the work. Thank you. Thank you, everybody. Actually, it was taken up for the diversity study actually. So very much less work has been done with the documentation of this Poaceae family itself, that is crab fishes. Very two work has been done actually. Previously, very long, that is, I told one in 1985 and one 1990 actually. One significance I can say, I can reveal here itself, that is whatever the documentation under 68, what I have species documented actually, under that 11 species have been not documented. They are new to this particular area, that is Kodagu district itself. They have to be upgraded, should be upgraded, the diversity species. Yeah, it's all the herbariums have been deposited in TUS Mysore University with the ocean. Thank you.
I take this privilege to welcome Dr. Ashok Banakar sir for this conference. Welcome. Sir. Next participant, Veena. Good evening. Myself, Veena. My topic title is Mapping of Seroprevalence of Leptospirosis among febrile patient suspected cases in Dakshina Kannada district, Karnataka, India 2020-21 under the guideship of Professor M. Jaishankar. Leptospirosis is a worldwide zoonotic disease and considered as a re-emerging disease caused by a bacteria, what is called as a Leptospira. It is endemic in the coastal region of India with frequent annual upsurges and outbreaks in the rainy season usually. It can infect various mammals and present as a febrile illness ranging from a mild subclinical infection to pulmonal multi-organ dysfunction in humans, similar to other hemorrhagic fevers like dengue fever. Dakshina Kannada coastal district of state Karnataka has been identified as one of the endemic area with increased cases of leptospirosis every year. The aim of the study is study of the leptospirosis seroprevalence in the clinically suspected cases, reporting to government health centers from 7 different taluka or blocks and private hospital, what is called as the Father Muller Hospital, Mangaluru of Dakshina Kannada district during 2021. And the objective has been taken as a mapping of seroprevalence of leptospirosis among the febrile patients suspected in Dakshina Kannada district, India 2020 and 21. The methodology which is involved as 1,621 serum samples have been taken for this. Clinical criteria has been taken as acute febrile illness of suspected leptospirosis cases. Samples involved is inpatient and outpatient visited to district and sub-district hospital of Dakshina Kannada district. As here it has been shown, the point map of distribution of the samples as more samples which has been taken from Mangalore Taluk. The methodology involved are enzyme linked immunosorbent test as well as a microscopic agglutination test. Leptospira IgM kit is used with J. Mitra and Company. Parallelly, all the sample has been run for the dengue also. Dengue IgM kit is National Institute of Virology kit has been used. Dengue NS1 antigen ELISA is J. Mitra and Company. ELISA was performed as per the manufacturer protocol. So out of this, the positives, 350 samples which have come positive, around 92 samples have sent for microscopic agglutination test to Leptospirosis Research Laboratory, ICR Nivedi, Bengaluru. A wide range of 20 Leptospira serovars covering 17 serogroups are involved under MAT. The results have analyzed by CISPR analysis, and we have found that leptospirosis distribution across the taluk was significantly associated with the leptospira IgM antibodies. As it could be seen, there are clustering of the cases in the 3 talukas like Mangaluru, Bandwala as well as Beltangadi. Total seroprevalence is 21.5% has been seen for leptospirosis. Parallel is run for all the samples for the dengue, which has been seen 9.7. It is also The analysis case for analysis has been done for year-wise, gender-wise in the age group, as well as a seasonal. In year-wise, gender-wise, as well as age group, it is not associated, as the p-value is seeing as more than 0.001, whereas it is less in case of seasonal, and it is in seasonal, it is more or less in the month of February, March, as well as in August and September, the cases are seeing high. Clinical symptoms are also not associated, as the p-value is more than 0.05, but most of the jaundice cases are seen in leptospirosis cases. The MAT result is more or less 44.6% is matching with the MAT, as the most of the cases are seen with the Putturu, Beltangadi, Putturu Sulya, Beltangadi, Bantwala, Kadaba, Mudabidre, and the least is Mangalore. Next. The serovar distribution is seen as these are the common serovars identified, as most of the serovar is Jasiman, Javanica, Panama, and Icteria hemorrhagiae, which has been commonly found in the coastal belt of Karnataka, is identified in Dakshina Kannada also. So at the end, the conclusion which would be taken as the GIS mapping, what has been done for the positive cases shows a clustering of urban populated area of different taluks of Dakshina Kannada. The present study provides insight into the leptospirosis prevalence in the endemic Dakshina Kannada, which will help to mitigate prevention of control strategies of management of leptospirosis in Dakshina Kannada district. Thank you.
Survey or clinical sample isolation, how is was carried out?
It is the sample which has come to the hospital has been taken for the survey purpose.
So did you see any genetical basis for the distribution of the disease? Genetical basis with patients?
No, sir. The clinical suspected febrile cases related to the leptospirosis, which is clinically diagnosed by the doctor, has been taken for the survey.
Okay. That's all.
Thank you, ma'am.
I thank oral presentation panel members, Professor Dayanand Akhtar sir and Dr. Jaikumar Nayan sir for judging this session. This oral presentation and poster presentation session will continue tomorrow also after technical sessions. Now I'll request everyone to rise for national anthem.
Keeping up with the pace of life. It's not easy to find the perfect balance. To see the rules. See, never ends.
I request everyone to attend cultural event. With shortly it will begin.
So please give your attention here. So at 6 o'clock, cultural program is going to start, and after that we'll be having dinner.